By 4 billion years ago, Earth had settled into relative calm. Oceans lapped against volcanic islands. The crust thickened into early continents. The sky glowed orange with organic haze. Lightning flickered across the warm atmosphere, and hydrothermal systems seethed along spreading ridges. Within this environment, chemistry took a decisive step toward biology. Carbon combined with nitrogen, hydrogen, and phosphorus to form complex molecules capable of storing information and replicating. The boundary between geochemistry and life blurred, and somewhere on this world of water and rock, the first living systems arose. All the clues about how life began are indirect. Scientists piece them together from ancient rocks, tiny fossils, and the chemistry of living cells. The oldest widely accepted signs of life come from rocks in Greenland that are about 3.8 billion years old, containing carbon that looks like it was processed by living things. Genetic evidence suggests that a single common ancestor of all life existed even earlier, around 4 billion years ago, meaning that life appeared surprisingly soon after the intense bombardment of Earth by asteroids had stopped. The young planet offered abundant raw materials. Volcanoes vented carbon dioxide, nitrogen, hydrogen, methane, and ammonia. Ultraviolet radiation, electric discharges, and geothermal energy provided the power to drive synthesis. In the atmosphere, lightning sparked reactions among these gases, while in the oceans, metal-rich vents catalyzed further complexity. The Earth was a vast chemical reactor. In 1953, scientists ran an experiment showing that sparks of electricity in a mix of simple gases could create amino acids, the building blocks of life. Later versions of the experiment, using gases like those from volcanoes, also produced other important ingredients such as acids and parts of genetic molecules. Today, researchers think Earth's early atmosphere wasn't as chemically rich everywhere, but in certain places, near volcanoes, lightning storms, or meteor impacts, the right conditions for life's chemistry could still have existed. Some meteorites, like the famous Murchison meteorite that fell in 1969, contain more than 80 different amino acids and other complex organic molecules similar to those found in living things. The chemical makeup of these materials shows they came from space. In Earth's early history, countless meteorites and comets brought huge amounts of this organic material, adding to, rather than replacing, the chemical building blocks already forming on Earth. Beneath the early oceans, tectonic and volcanic activity produced networks of submarine vents. At mid-ocean ridges, seawater percolated into hot basalt, reacted with minerals, and emerged as plumes laden with hydrogen, methane, and sulfides. Where these fluids mixed with cold seawater, minerals precipitated into chimneys, creating steep chemical and thermal gradients, ideal environments for complex reactions. Today's deep-sea vents come in different types. Some, called black smokers, are extremely hot and rich in metals. Others, like the Lost City vents in the Atlantic, are cooler and alkaline. These form when certain rocks react with seawater, releasing hydrogen and creating tiny natural chambers with chemical energy. Inside these spaces, simple carbon molecules such as formate, acetate, and methane can form, the same kinds of molecules that living cells use for energy and growth. Lab experiments have shown that thin layers of iron and nickel sulfides can help spark the creation of simple energy-rich molecules, the kind that may have powered early life. These reactions resemble those used in one of the most ancient ways living cells make energy today. In other words, the chemistry around hydrothermal vents may have provided both the building blocks and the energy systems that early life needed to begin. Studies of ancient sulfur minerals show chemical patterns that suggest microbes were already using sulfur for energy more than 3.4 billion years ago. These patterns match what we'd expect from life that developed around hydrothermal vents, using the natural chemical differences between hot, hydrogen-rich fluids and cooler, sulfate-rich seawater to power its metabolism. At some point, geochemical complexity gave rise to molecules capable of both storing information and catalyzing reactions. Ribose nucleic acid, RNA, can serve both functions. It can encode sequences and fold into catalytic shapes known as ribozymes. The RNA world hypothesis proposes that before DNA and proteins, life was based on RNA self-replication within confined microenvironments such as mineral pores or lipid vesicles. Experiments have shown that activated nucleotides can polymerize on clay minerals such as montmorillonite, forming RNA chains tens of bases long. RNA molecules can evolve new catalytic activities through iterative selection in vitro, including self-ligation and partial replication. The modern ribosome, a ribozyme that catalyzes peptide bond formation, is considered a living fossil of this early era. The main difficulty lies in prebiotic synthesis of ribose and nucleobases under plausible early Earth conditions. Recent studies using ultraviolet irradiation of cyanide-derived intermediates yield simultaneous formation of ribonucleotides, suggesting that an integrated photochemical pathway could have operated on the early Earth surface. Genetic studies suggest that all life today shares a single ancient ancestor, a simple organism that used RNA-based enzymes and only a few basic proteins. This ancestor likely gained energy by reacting hydrogen with carbon dioxide and by fixing nitrogen, much like some microbes found around hydrothermal vents today. These similarities hint that life's first systems may have grown directly from the chemistry of the early Earth. Other hypotheses complement or compete with the RNA world. The metabolism-first model posits self-sustaining chemical cycles, such as iron-sulfur catalysis, that predated genetic polymers. The lipid world suggests that amphiphilic molecules spontaneously formed vesicles enclosing reaction networks. Panspermia envisions microbial life arriving from space on meteorites, a mechanism demonstrated possible but not necessary. Current consensus holds that multiple processes operated together. Geochemical energy supplied raw organics, self-assembling membranes provided compartments. RNA-like molecules arose within these niches and gradually evolved heredity. By 3.9 billion years ago, the prebiotic Earth had crossed a threshold. Chemical systems capable of replication and metabolism began to outcompete purely abiotic processes. Within the sheltered niches of hydrothermal vents, clay flats, and tide pools, microscopic compartments formed, droplets, vesicles, or mineral pores enclosing catalytic cycles. Natural selection, operating at the molecular level, began to sculpt complexity. The first true cells appeared, small, anaerobic organisms feeding on the chemical energy of their environment. Studies of modern genetics show that every living thing on Earth can be traced back to a single ancient ancestor that lived about 4 billion years ago. This organism, called Luca, probably had only a few hundred genes, just enough to let it make energy from hydrogen, turn carbon dioxide into organic molecules, and process nitrogen. It likely lived around hydrothermal vents, using natural metal-rich minerals and chemical gradients to power its life processes, much like some vent-dwelling microbes do today. Molecular clocks calibrated against the fossil record place the divergence of bacteria and Archaea near 3.9 Ga, implying that life's diversification followed swiftly after its origin. This rapid appearance supports a model where habitability and biogenesis coincided soon after planetary stabilization. As life spread beyond vents, microbial communities colonized shallow waters. Layers of cyanobacteria and other microbes trapped sediments, forming laminated structures known as stromatolites. These living carpets represented one of the earliest large-scale interactions between biology and geology. Through photosynthesis, they began to alter the chemistry of oceans and atmosphere. The oldest confirmed stromatolites, layered structures built by microbes, are found in Western Australia's Pilbara region and South Africa's Barberton Mountains, both about 3.5 billion years old. Their rounded shapes, fine layers, and chemical fingerprints show that they were formed by living organisms, not just natural mineral processes. Even older possible examples from Greenland, dating to around 3.8 billion years ago, are still debated but hint that microbial life using sunlight or chemical energy may have begun even earlier. Stromatolites form as microbial films secrete sticky extracellular polymers that trap carbonate or silicate particles. Successive generations grow atop one another, producing microlaminae 0.1 to 1 mm thick. These accretionary structures provide the earliest macroscopic fossils of life, visible in outcrop even billions of years later. At first, most organisms were chemotrophs, gaining energy from reactions between hydrogen, methane, and sulfides. But at some point, microbes learned to harness sunlight. Primitive anoxygenic photosynthesis used iron or hydrogen sulfide as electron donors, yielding elemental sulfur or ferric oxides instead of oxygen. Later, a new lineage, ancestors of modern cyanobacteria, evolved to use water itself as the electron donor, releasing O as a byproduct. This innovation transformed the planet. Genetic studies suggest that microbes capable of making oxygen through photosynthesis evolved very early, perhaps as far back as 3.5 billion years ago. Ancient stromatolites from that time already show traces of rust-like iron minerals, evidence that oxygen was being produced in small amounts. The key proteins that drive photosynthesis appear to have split from common ancestors deep in Earth's early history. By about 2.9 billion years ago, chemical clues in rocks show that oxygen was starting to escape into the air and react with minerals on land. The biochemical trick was the evolution of the MnCaO cluster within photosystem II, capable of splitting water into protons, electrons, and oxygen. Each molecule of O liberated by these microscopic engines carried profound geochemical consequences. Iron dissolved in seawater oxidized to rust, forming banded iron formations that now line ancient cratons. The ratio of carbon types in ancient rocks gives one of the clearest clues that life has existed on Earth for billions of years. Living things prefer to use the lighter form of carbon, which leaves a distinct chemical fingerprint behind. In very old rocks, minerals show a mix of heavier carbon, while the carbon from ancient organic matter is much lighter, a difference that can only be explained by life. This pattern appears as far back as 3.8 billion years ago, showing that Earth's carbon cycle, driven by living organisms, has been running almost since the planet began. By 3.5 Ga, the Earth supported a global microbial biosphere. Oceans teemed with bacterial and archaeal life, cycling carbon, sulfur, and nitrogen. Methanogens produced methane, maintaining a greenhouse that compensated for the faint sun. Iron and sulfur oxidizers colonized hydrothermal settings, while photosynthetic mats spread across continental shelves. Life had become a planetary force, altering atmospheric composition and sediment chemistry. Sedimentary porites from 3.47 Ga show multiple sulfur isotope anomalies pointing to microbial sulfur cycling under an anoxic sky. Nitrogen isotopes from the same strata indicate biological nitrogen fixation via the molybdenum-dependent nitrogenase enzyme, demonstrating that key biochemical pathways were already ancient. Together, photosynthetic microbes and methane-producing organisms helped keep early Earth warm. Their activity released both carbon dioxide and methane into the air, creating a natural greenhouse that prevented the planet from freezing. Sunlight broke down some of the methane, forming a thin orange haze, much like the one around Saturn's moon Titan, which blocked harmful ultraviolet rays while still trapping enough heat to keep the planet comfortable. The gradual rise of oxygen did not yet transform the atmosphere. Iron and sulfur sinks absorbed most O. But by about 2.5 Ga, these buffers would saturate, triggering the Great Oxidation Event. That later transformation belongs to the Proterozoic, but its foundations were laid here in the Archean seas, in the humble metabolic experiments of stromatolites and cyanobacteria that learned to turn light and water into power. Life's influence on the planet had begun its long ascent. Minerals like pyrite and uraninite, which can only exist where there's little or no oxygen, vanished from river deposits about 2.4 billion years ago. At the same time, red-stained rocks and unusual sulfur signatures began to appear, clear signs that oxygen had finally started to build up in the air. This marked the turning point from Earth's oxygen-free past to a new, more oxidized world. The first sparks of life were not a single event but a continuum, from minerals to molecules, from metabolism to replication, from chemistry to heredity. Within a few 100 million years of planetary calm, Earth changed forever. Self-organizing systems began to evolve, reproduce, and reshape their environment. By the end of this episode, life was established across the planet, preparing for the next great leap, the oxygenation of the atmosphere and the flowering of complex cells that would follow in the Proterozoic world. For more than 1 billion years after life began, Earth's atmosphere remained devoid of free oxygen. The oceans were green with dissolved iron and sulfides. The sky likely retained a reddish haze of methane. Then, about 2.4 billion years ago, this equilibrium collapsed. Oxygen released by microbes accumulated faster than geological sinks could absorb it. Within a geological instant, the chemistry of air, water, and rock changed. The planet rusted, the haze cleared, and for the first time Earth possessed an oxidizing atmosphere. This transformation, the Great Oxidation Event, set the stage for all complex life to come. Scientists can trace Earth's changing atmosphere through chemical clues in ancient rocks. 1. Before 2.7 billion years ago, the oceans had almost no oxygen and were rich in dissolved iron. Minerals like pyrite and uraninite could survive in river sediments because the air and water were oxygen-free. 2. Between about 2.5 and 2.3 billion years ago, things began to change. Those oxygen-sensitive minerals disappeared, and rusty red rocks started to form on land, evidence that oxygen was appearing in the environment. Sulfur atoms in rocks also began showing unusual patterns that point to a shifting atmosphere. 3. After 2.3 billion years ago, oxygen became a permanent part of the air, though still only a tiny fraction of today's level. This major shift, called the Great Oxidation Event, happened around 2.32 billion years ago, marking the first time Earth's atmosphere held a lasting supply of oxygen. Long before oxygen filled the air, it was being produced in the sea. Cyanobacteria, microscopic architects of stromatolites, had perfected oxygenic photosynthesis. Using sunlight, water, and carbon dioxide, they generated organic matter and released oxygen as waste. For hundreds of millions of years that oxygen reacted immediately with reduced iron, sulfur, and volcanic gases. Only when these sinks became saturated could molecular oxygen escape into the atmosphere. The photosynthetic reaction. Photosynthesis, the process where sunlight turns carbon dioxide and water into sugar and oxygen, can be summed up by the equation: 6 molecules of carbon dioxide plus 6 molecules of water, in the presence of light, produce 1 molecule of glucose and 6 molecules of oxygen. This means that for every unit of carbon taken in, 6 units of oxygen are released. Today, mats of cyanobacteria, tiny photosynthetic microbes, can produce around 10 grams of oxygen per square meter each day. If similar microbial communities had covered the shallow seas of the early Earth, they could have filled the entire atmosphere with oxygen in just about 10 million years, if none of it had been used up. But oxygen didn't build up that quickly. It actually took more than 300 million years, because vast amounts of it were being consumed by chemical reactions with iron and other elements in the oceans and crust. These acted as oxygen sinks, soaking up the gas long before it could accumulate in the air. Molecular fossils known as 2-methylopanes, preserved in 2.7 billion-year-old shales from the Pilbara Craton, record cyanobacterial lipids predating the GOE. These biomarkers confirm that oxygenic photosynthesis evolved well before atmospheric oxygenation. Isotopic fractionations in sulfur and iron likewise show local molecular oxygen oases in surface waters hundreds of millions of years prior to the global shift. Once these sinks filled, oxygen levels began to rise exponentially. Iron in the oceans precipitated as ferric oxides, turning the seas from green to red. Methane, previously abundant, was oxidized to CO, collapsing the greenhouse and cooling the planet. Ultraviolet light acting on oxygen produced ozone, forming the first stratospheric shield. The air itself transformed from reducing to oxidizing, changing every mineral at the surface and every metabolic pathway in living things. Rocks from about 2.3 billion years ago show the first appearance of red beds, sandstone stained by the rusty mineral hematite. This tells us that iron was reacting with oxygen on land, forming the red color we still see today. At the same time, strange patterns in sulfur isotopes vanished from the rock record. These patterns only form when there's almost no oxygen in the air, so their disappearance shows that oxygen had begun to spread through the atmosphere, reaching at least 100,000th of today's level. Other chemical clues, such as the ratios of chromium and molybdenum isotopes, reveal that rain and rivers had started to weather continental rocks in the presence of oxygen as early as 2.48 billion years ago. Together, these different lines of evidence mark one of the most important turning points in Earth's history, the rise of atmospheric oxygen. The rise of oxygen completely changed the course of life on Earth. Microbes that couldn't tolerate oxygen, called anaerobes, were forced to survive in oxygen-free environments, while new species evolved that could use oxygen to gain energy. The breakthrough was aerobic respiration, a process where living things use oxygen to break down sugar and release energy. In words, the reaction goes like this. 1 molecule of glucose plus 6 molecules of oxygen produces 6 molecules of carbon dioxide, 6 molecules of water, and a large amount of energy. This new way of releasing energy was incredibly efficient, about 18 times more powerful than the old method of fermentation. It was a biochemical revolution that eventually made complex, multicellular life possible. The most striking evidence of Earth's first oxygen boom is found in banded iron formations, or BIFs, ancient seafloor rocks made of alternating layers of silica, chert, and iron oxide that formed between 2.7 and 2.4 billion years ago. In the early oceans, which had no oxygen, iron existed in a dissolved form that made the water rich and murky. When photosynthetic microbes began releasing oxygen, that oxygen reacted with the iron, turning it into solid rust-like minerals such as hematite and magnetite. These minerals sank to the seafloor, forming thick, layered deposits. Over time, this process removed more than 100 trillion tons of iron from seawater, effectively trapping much of the new oxygen inside rock and giving the ancient crust its deep red hues. The disappearance of BIFs after about 2.4 billion years ago shows that most of the iron in the oceans had finally been oxidized, and the seas were starting to clear. The rise of oxygen, while a triumph for emerging life, unleashed profound climatic consequences. Methane, previously the dominant greenhouse gas, reacted rapidly with oxygen to form carbon dioxide and water. Because carbon dioxide is less efficient per molecule at trapping heat, the planet's surface temperature fell sharply. Within a few million years of the atmospheric transition, glacier advanced toward the equator. Earth entered what may have been its first global ice age, a snowball Earth. In an oxygen-free world, methane could linger in the atmosphere for around 10,000 years, acting as a powerful greenhouse gas that helped keep the planet warm. But once oxygen levels rose to even about 1% of today's amount, methane began to break down much more quickly, surviving less than a decade. As a result, the amount of methane in the air plummeted, and Earth lost a major source of heat. This drop in greenhouse warming likely cooled the planet by more than 20 degrees Celsius, triggering global ice ages that left glacial deposits even in what were once tropical regions. Clues to the great ice ages of the early Earth come from ancient glacial rocks, mixtures of pebbles, mud, and large dropstones left behind by melting ice. These have been found in places like Canada, South Africa, and Finland, dating to around 2.4 to 2.2 billion years ago. Magnetic data show that some of these icy deposits formed within 15 degrees of the equator, suggesting that glacier once reached nearly every part of the planet. At the same time, chemical signals in ancient limestone, unusually high levels of the carbon isotope to the power of 1 cubed C, reveal that huge amounts of organic carbon were buried underground. This process would have released even more oxygen into the atmosphere, reinforcing one of the most dramatic environmental shifts in Earth's history. During this interval, roughly 2.4 to 2.2 billion years ago, Earth may have been locked in ice from pole to equator. Oceans froze over, sea ice kilometers thick smothered the surface, and sunlight reflected back into space. Yet volcanic outgassing continued beneath the ice, releasing CO faster than weathering could remove it. After millions of years, greenhouse gases accumulated, triggering a catastrophic thaw. The ice melted in geologic days, the oceans boiled with super greenhouse temperatures, and chemical weathering of the exposed continents reset the carbon cycle. Computer models of ancient climates suggest that if Earth's surface reflected more than 60% of incoming sunlight, global temperatures could have dropped below -50 degrees Celsius, plunging the planet into a deep freeze. Escaping such a snowball Earth would have required enormous amounts of carbon dioxide, more than 100,000 parts per million, hundreds of times higher than today's levels, to trap enough heat for the ice to melt. When the world finally thawed, this dramatic warming left behind telltale layers of cap carbonates, thick deposits of dolomite and limestone laid down quickly on top of glacial rocks, marking the sudden shift from a frozen planet to a sweltering greenhouse world. Chemical clues within these cap carbonates, particularly high levels of the carbon isotope to the power of 1 cubed C, show that huge amounts of buried organic carbon were rapidly oxidized as the planet warmed after the great ice ages. This process released vast quantities of carbon dioxide back into the atmosphere and oceans, driving a powerful rebound in greenhouse conditions. The event not only transformed Earth's climate but also changed the way the global carbon cycle interacted with life, making the planet's atmosphere and climate far more responsive to biological activity from that point onward. Oxygenation did more than freeze and thaw the planet. It transformed mineralogy, oceans, and evolution itself. Over 2,500 new minerals, oxides, sulfates, and carbonates appeared for the first time, giving Earth its characteristic palette of reds, greens, and blues. The chemistry of the oceans shifted from iron-rich to sulfur-rich, paving the way for new microbial metabolisms. Aerobic respiration evolved, providing an enormous energy advantage and setting the stage for complex cells. Computer models of ancient oceans show that after the Great Oxidation event, Earth seas developed a layered structure. The surface waters contained oxygen, the middle layers had little oxygen but were rich in sulfur compounds, and the deepest parts remained rich in dissolved iron and completely oxygen-free. This pattern lasted for nearly 1 billion years, a period often called the boring billion, when early complex cells, or eukaryotes, existed but struggled to thrive because the oceans lacked enough nutrients. Over time, as iron was permanently removed from seawater, nutrients like phosphate began to combine with calcium instead. This gradual change enriched the upper ocean with the ingredients needed to support more abundant and complex life. Evidence from ancient minerals confirms this great environmental shift. Before about 2.4 billion years ago, certain minerals like uraninite and pyrite could survive in river sediments because there was no oxygen to destroy them. After oxygen began to accumulate, these minerals disappeared and were replaced by rust-colored iron oxides such as hematite and goethite. along with sulfate-rich salts that formed as shallow seas evaporated. The rise of oxygen also caused sulfur-bearing rocks on land to break down more easily, sending sulfate into the oceans. This new supply of sulfur supported a variety of microbes that used it for energy, marking another key step in Earth's evolving biosphere. The GOE was both a catastrophe and an opportunity. For anaerobes, oxygen was toxic. It attacked cellular machinery and generated reactive radicals. Many lineages retreated underground or to anoxic sediments, giving rise to modern anaerobic clades. But a few adapted. Enzymes such as superoxide dismutase and catalase evolved to neutralize reactive oxygen species, allowing new forms of metabolism to thrive. Some microbes turned the poison into power, developing aerobic respiration, nitrification, and sulfur oxidation. These innovations increased metabolic yield by an order of magnitude and prepared the biochemical toolkit that later supported multicellular life. Comparative genomics shows that the genes encoding these antioxidant enzymes are ancient, with duplication and diversification occurring near the 2.4 billion year horizon. Molecular clocks for cytochrome oxidases, the terminal components of aerobic respiration, also cluster in this window, linking molecular evolution directly to atmospheric transformation. By about 2.0 Ga, oxygen levels stabilized at roughly 1 to 10% of present atmospheric levels. The Earth had entered the Proterozoic eon. The sky turned blue under the newly formed ozone layer. The oceans cleared. Continental weathering accelerated. The climate oscillated between mild warmth and deep freeze, but the biosphere endured. Life had survived its first planetary crisis and emerged stronger, armed with the tools to colonize an oxidizing world. After the Great Oxidation Event, the amount of carbon dioxide in the atmosphere dropped sharply as oxygen-rich weathering of rocks began again. In the oceans, levels of sulfate, a compound formed when sulfur reacts with oxygen, increased, while the deepest waters remained low in oxygen and rich in dissolved iron. This partly oxygenated world persisted for more than 1 billion years, a long era of balance between old and new chemical conditions. Eventually, another major rise in oxygen, known as the Neoproterozoic oxygenation event, transformed the planet once again, creating the environmental conditions that allowed animals to evolve. The Great Oxidation Event was Earth's first biological revolution. A gas exhaled by humble microbes remade the planet, froze the seas, and rusted the continents. It marked the end of the Archean world and the beginning of the Proterozoic, a planet breathing for the first time. Oxygen would never again vanish. Its concentration would fluctuate, but its presence would define the character of all later evolution. In the next episode, the focus turns inward to the microscopic frontier, the rise of eukaryotic cells and the dawn of complex life.
How sex changed everything, the secret revolution of complex life. Part 1, The Redox World of the Mid-Proterozoic. Introduction, A Planet in Suspension.
Between two cataclysms, the Great Oxidation of 2.4 billion years ago and the Cryogenian glaciations around 720 million, stretches a long, still epoch. Continents weathered slowly beneath a muted sun. Oceans lay stratified beneath sluggish atmospheres. It has been called the boring billion, yet within this quietness life achieved one of its most extraordinary transformations. Bacteria and Archaea, the only forms of life for 2 billion years, joined in a partnership that created the first complex cells, the eukaryotes. Within their membranes lay the seeds of all later animals, plants, and fungi. Between about 1.8 and 0.8 billion years ago, Earth entered a long period of stability in both climate and oxygen levels. The atmosphere contained only a small fraction of today's oxygen, enough for simple, oxygen-breathing microbes to survive, but not enough to support large or complex animals. In the oceans, the surface waters held some oxygen, but the deeper layers remained dark and rich in dissolved iron, with little or no oxygen at all. This chemical layering created both challenges and opportunities for life, driving the evolution of new ways for organisms to harness energy and adapt to their changing world.
The redox world.
The Proterozoic oceans were unlike today's blue expanses. Beneath a thin oxygenated skin lay vast green depths loaded with dissolved iron. Sunlight penetrated only the upper few tens of meters. Below, sulfate-reducing bacteria thrived in dark, sulfur-rich waters. Periodic mixing brought trace metals and nutrients upward, feeding photosynthetic plankton. These sluggish chemical cycles formed the crucible in which complex cells evolved. Chemical clues locked in ancient rocks helped scientists understand what Earth's environment was like during this time. After about 2 billion years ago, certain metals such as molybdenum and uranium became more concentrated in ocean sediments, showing that parts of the oceans were starting to gain oxygen. Other elements, like chromium and cerium, reveal that oxygen levels still rose and fell over time. Meanwhile, zinc, a metal vital for building DNA and proteins, became about 3 times more abundant between 2.1 and 1.5 billion years ago, matching the appearance of the first complex, eukaryotic cells in the fossil record. These metals were crucial for key biological processes such as breathing and cell division, showing how changes in the chemistry of the oceans helped pave the way for more complex life. Around 1.8 billion years ago, the chemistry of Earth's oceans began to shift. Waters that had once been rich in dissolved iron became increasingly filled with sulfur compounds, creating what scientists call sulfide-rich or euxinic conditions. This change had major consequences for life. Sulfide tends to lock away important metals like molybdenum and copper, which living things need for vital processes such as breathing and capturing nitrogen from the environment. With these metals in short supply, life entered a kind of nutrient drought, a long period when evolution slowed dramatically. This scarcity may help explain why, for nearly 1 billion years, the pace of biological innovation appeared to stall, a time scientists refer to as the boring billion.
The Archaeal Foundation.
Modern molecular phylogenies reveal that eukaryotes did not arise from typical bacteria but from within the Archaea, a domain of heat-tolerant microbes inhabiting vents and sediments. Somewhere in the mid-Proterozoic ocean floor, an archaeal host cell formed intimate contact with a bacterium capable of using oxygen. What began as ingestion or parasitism became symbiosis. The bacterium persisted inside the host, gradually transforming into an organelle, the first mitochondrion. The host lineage belonged to the Asgard Archaea, discovered in 2015 from deep-sea sediments. Their genomes encode actin-like and tubulin-like proteins, small GT parses, and ESCRT complexes, molecular machinery once thought unique to eukaryotes. These genes provide the missing evolutionary bridge between simple prokaryotes and nucleated cells. Rising oxygen levels around 1.9 billion years ago created the right conditions for one of life's most important breakthroughs, the merger that gave rise to complex cells. Geological evidence, including the appearance of red-colored rocks and changes in sulfur isotopes, shows that oxygen was spreading across the planet at this time. In shallow coastal waters, small pockets of oxygen, or oxygen oases, formed near the edges of continents. Here, oxygen-using bacteria and oxygen-hating Archaea could live side by side, coming into close contact again and again. Eventually, one of these encounters led to a lasting partnership, endosymbiosis, in which a bacterial cell took up residence inside an archaeal host. This relationship could only succeed once there was enough oxygen in the environment for the bacterial partner to provide real energy benefits, setting the stage for the evolution of complex life.
The oldest eukaryotic fossils.
The fossil record of this transformation is sparse but tantalizing. Spherical, ornamented microfossils with complex wall structures appear in shales of the Grapania spiralis assemblage at 1.87 billion years. Thread-like coils up to 2 mm long, the first truly macroscopic organisms. Later forms such as Topania plana, 1.63 billion years, show branching extensions suggesting cytoskeletal organization and possible colony formation. Tiny fossils called acritarchs provide some of the earliest evidence for complex, eukaryotic life. These microscopic spheres, ranging from 20 to 300 micrometers across, have tough outer walls and internal structures that closely resemble those of modern algae rather than bacteria. Under powerful electron microscopes, their walls show a distinctive double-layered design made of durable organic compounds found only in eukaryotes. Chemical signatures within these fossils also point to photosynthesis, the process of using sunlight to make food. Because acritarchs have been discovered on several continents, scientists know that by about 1.6 billion years ago, these early complex cells had already spread across the globe. Chemical traces found in ancient rocks add powerful support to the fossil evidence for early complex life. In rocks from the Barney Creek Formation in Australia, dating to about 1.64 billion years ago, scientists have discovered molecules called steranes. These are the preserved remains of sterols, fats that help strengthen the cell membranes of eukaryotes, the same type of cells that make up plants, animals, and fungi today. Importantly, making sterols requires oxygen, so their presence not only confirms that eukaryotic life had evolved by this time, but also shows that oxygen was already present in the surface waters where these organisms lived.
Transition summary.
By 1.6 billion years ago, the basic blueprint of the eukaryotic cell was established: a nucleus protecting DNA, a cytoskeleton for shape and motion, and mitochondria generating internal power. The merger of archaean and bacterium had produced an organism capable of complexity far beyond its ancestors. The stage was set for further symbioses, and for the invention of sex and multicellularity.
Part 2: Energy, Symbiosis, and the Birth of Organelles. The energy barrier.
Life's potential for complexity is limited by energy. Bacteria and archaea, though immensely diverse, face a physical constraint. They generate ATP only across their outer membranes. The surface area of that membrane scales far more slowly than the cell's internal volume, and beyond a certain size diffusion can no longer deliver enough energy. For nearly 2 billion years, this law kept cells microscopic. The evolution of eukaryotes broke it. Energy models show that simple cells, like bacteria, face a hard limit on how large and complex they can become. Once a cell grows beyond a certain size, it no longer has enough surface area to produce the energy needed to run its internal machinery or maintain a large genome. The invention of mitochondria changed everything. These tiny, energy-producing compartments acted like thousands of miniature power stations inside a single cell. A typical eukaryotic cell could contain thousands of mitochondria, each generating its own supply of energy. This massive boost, up to 20,000 times more energy-producing surface area than before, allowed cells to expand their DNA, build complex internal structures like the cytoskeleton, and develop the sophisticated systems of communication and control that made multicellular life possible. Around 1.9 billion years ago, the rise of oxygen gave life access to a far more powerful source of energy. Organisms that learned to use oxygen in their metabolism could extract roughly 15 times more energy from the same amount of food than those relying on older, oxygen-free processes. Even though oxygen made up only a tiny fraction of today's levels, it was enough to revolutionize the way cells produced energy, a change that transformed life's entire biological economy and paved the way for greater complexity.
Endosymbiosis, the mitochondrial union.
At some point, an archaeal host engulfed, but did not digest, an alpha proteobacterial partner. The bacterium brought the machinery for oxidative phosphorylation. The host supplied protection and nutrients. Over generations, genes migrated from the symbiont's genome to the host's nucleus, tying their fates together. The symbiont shrank into an organelle, the mitochondrion. Genetic studies show that modern mitochondria, the tiny powerhouses inside our cells, still carry a handful of their own genes, usually between 37 and 70, compared with the roughly 2,000 genes their free-living bacterial ancestors once had. Over time, most of those genes were transferred to the cell's main nucleus, allowing the host cell to take direct control of energy production and coordinate its metabolism more efficiently. Despite the wide variety of species that exist today, all mitochondria share more than 50 essential proteins, pointing to a single common origin from an ancient symbiotic event about 1.85 billion years ago, when one cell permanently merged with another. The host archaean probably lived in anoxic sediments at the oxic-anoxic interface. In such gradients, hydrogen-producing bacteria and hydrogen-consuming archaea already formed syntrophic consortia. Endosymbiosis may have begun as a permanent hydrogen transfer partnership, the hydrogen hypothesis, stabilized by gene exchange and eventual internalization. Chemical clues from rocks about 1.64 billion years old reveal clear signs of a major biological transition. In these ancient shales, scientists have found molecules such as steranes, squalenes, and hopanoids, chemical fossils that together indicate thriving communities where bacteria produced organic matter and early eukaryotes consumed it. At the same time, changes in the chemical makeup of iron within nearby sediments show that more of it had reacted with oxygen, a pattern consistent with the spread of aerobic respiration, the process of using oxygen to release energy from food. Together, these signals show that oxygen was reshaping both Earth's chemistry and its living ecosystems.
The birth of chloroplasts.
Hundreds of millions of years after mitochondria, a second endosymbiosis occurred. A heterotrophic eukaryote engulfed a cyanobacterium capable of oxygenic photosynthesis. Instead of digesting it, the host retained it as a living light-harvesting organelle, the chloroplast. This event founded the lineage of algae and, later, plants. The DNA inside chloroplasts, the structures in plant and algal cells that carry out photosynthesis, is strikingly similar to that of modern cyanobacteria, particularly filamentous types resembling nostoc. Genetic comparisons show that only about 5 to 10% of chloroplast genes still match those in living cyanobacteria. The rest were gradually transferred to the host cell's nucleus over time. The oldest known photosynthetic eukaryotes, the red and green algae, had already split into separate groups by about 1.6 billion years ago, meaning the original partnership, when a cyanobacterium became permanently trapped inside another cell to form the first chloroplast, must have happened slightly earlier. Chemical traces found in rocks from the 1.1 billion-year-old Xiamling Formation in northern China provide strong evidence that early eukaryotes were already performing oxygen-producing photosynthesis. These rocks contain molecules such as phytane and pristane, which come from chlorophyll A, the same pigment used by modern algae and plants to capture sunlight. The appearance of these compounds also lines up with a rise in phosphate and molybdenum in the oceans, two key nutrients that help drive biological growth. Together, these clues show that by this time, algae were thriving, and Earth's oxygen cycle was becoming increasingly influenced by eukaryotic life. Later, secondary and tertiary endosymbioses multiplied photosynthetic diversity. Eukaryotes such as diatoms, dinoflagellates, and euglenids captured whole algal cells, creating nested organelles with 3 or 4 membranes. This proliferation of plastids transformed marine ecosystems and, eventually, atmospheric composition.
The architecture of complexity.
Mitochondria and plastids were only part of the story. Within eukaryotic cells, a dynamic cytoskeleton of actin and microtubules enabled internal transport, phagocytosis, and cell division. Vesicles budded from endomembranes, giving rise to the Golgi apparatus and endoplasmic reticulum. The nucleus isolated transcription. Chromosomes wrapped around histons and recombined through meiosis. The cell had become a community within itself. The development of the cytoskeleton, the internal framework that gives cells shape and movement, was a major step in the evolution of complex life. Key proteins such as actin and tubulin, which form the filaments and tubes inside modern cells, can be traced back to ancient relatives found in a group of microbes called Lokiarchaeota. When these proteins link together, they create flexible structures that allow cells to move, change shape, and even engulf food particles, a process known as phagocytosis. This ability to take in nutrients and form close partnerships with other cells set the first eukaryotes apart from simpler, rigid-walled microbes, paving the way for greater complexity. Some of the first large microscopic fossils of complex cells, including forms known as Topania, Valeria, and Arcticellularia, show intricate surface patterns that suggest they already had an internal cytoskeleton to shape and support their cell walls. These fossils, dated to between 1.63 and 1.45 billion years ago, appear at the same time as chemical evidence showing a rise in biological activity and oxygen levels in the oceans. Shifts in the carbon isotopes from this period point to more active photosynthesis and carbon cycling, indicating that Earth seas were becoming increasingly rich in life.
The eukaryotic expansion.
By 1.4 billion years ago, eukaryotic life had spread through coastal oceans. Diverse protists, amoebae, flagellates, early algae, inhabited microbial mats and planktonic communities. They fed on bacteria, recycled nutrients, and oxygenated the photic zone. Though invisible to the macroscopic world, they began to reshape the biosphere from the bottom up. Between 1.6 and 1.2 billion years ago, the rate at which organic carbon was buried in sediments rose by about 30%, according to chemical evidence from ancient rocks. This means that living organisms were becoming more effective at trapping carbon in the deep ocean and seafloor, a process sometimes called the biological pump. As more carbon dioxide was locked away, oxygen levels in the atmosphere rose slightly. This long-term feedback between life and the environment helped create the conditions that would eventually allow multicellular animals to evolve. The geologic record captures the spread of these ecosystems in laminated shales and black mudstones rich in organic matter. Biomarkers include steranes, hopanes, and isoprenoids indicating both producers and heterotrophs. These strata underpin the mid-Proterozoic sedimentary quietude, tectonically calm, biologically steady, yet slowly oxygenating.
Transition summary.
Through endosymbiosis and internal reorganization, eukaryotes solved the energy problem and invented cellular architecture. Within them, life became compartmentalized, powered, and capable of innovation. The quiet billion years that followed were not barren stagnation but the gestation of complexity itself.
Part 3. The invention of sex and early multicellularity. The invention of sex.
Complex cells brought with them an opportunity unknown to the microbial world, the ability to combine, exchange, and reshuffle genes. Somewhere between 1.6 and 1.2 billion years ago, eukaryotes evolved meiosis, the process that halves chromosome numbers and allows gametes to fuse. Through sexual reproduction, genetic information could be recombined each generation, producing endless variation for natural selection to act upon. The world's first sexual lineages appeared long before animals, flowers, or spores. Modern genetic studies show that all major groups of complex life, including animals, plants, fungi, and single-celled protists, share the same set of core genes that control sexual reproduction. These genes, which guide the process of meiosis, the division of cells to form sperm and eggs, are so similar across all eukaryotes that scientists believe sex evolved only once, in the common ancestor of all complex life. Molecular dating of these genes places this breakthrough between 1.6 and 1.4 billion years ago. This timing matches geological evidence showing higher levels of key metals like zinc and copper, which these enzymes require, and signs of increased nutrient recycling in ancient marine sediments, a sign that Earth's ecosystems were becoming more dynamic and interconnected. The key advantage of sexual reproduction is that it allows genes to be reshuffled each generation, creating new combinations that help species adapt. In contrast, organisms that reproduce asexually simply copy their DNA, which means harmful mutations can build up over time, a process known as Muller's ratchet. By mixing genetic material from two parents, sex effectively resets the genome, repairing damage and introducing useful variation. Computer models show that for small organisms like early eukaryotes, which typically lived in populations of about 100,000 to a million individuals, sexual reproduction could double the rate of beneficial genetic change, even though it required more effort and produced fewer offspring overall. The earliest clear evidence of sexual reproduction in the fossil record comes from a species called Bangiomorpha pubescens, found in rocks in Arctic Canada that are about 1.05 billion years old. These fossils belong to a type of red algae made up of thin, thread-like filaments. Within them, scientists have identified structures that look like spores and reproductive organs known as gametangia, showing that the organism alternated between two life stages, one with a single set of genes and one with a double set. This discovery marks the oldest known example of both sex and multicellularity in the same organism, representing a major milestone in life's evolution.
The road to multicellularity.
Life's next great leap was cooperation. Once cells could adhere and communicate, they could divide labor, forming structures larger than any individual. The earliest experiments in multicellularity likely occurred many times and independently, in red and green algae, fungi, and protoanimals. At first, these colonies were simple filaments or spheres, held together by extracellular polymers. Later, they developed specialized tissues. The genetic foundations for multicellularity predate its appearance. Comparative genomics reveals that unicellular choanoflagellates and vulvacine algae already possess adhesion, cadherin-like, signaling, tyrosine kinase, and differentiation, transcription factor, genes homologous to those in animals and plants. These genes were co-opted from ancestral roles in cell-cell recognition and prey capture. Fossils of multicellular life older than 1 billion years show that complex organisms appeared far earlier than once thought. One striking example is Gabonionta, found in 2.1 billion-year-old rocks in Gabon, Africa. These fossils are a few centimeters across and have lobed, organized structures that suggest the cells grew and developed together in a coordinated way. Although scientists are still unsure exactly what kind of organisms they were, these fossils prove that multicellular organization evolved long before the famous Cambrian explosion. Later examples, such as Bangiomorpha, a red alga about 1 billion years old, and Paleastrum, a green alga about 900 million years old, show that multicellularity arose more than once, especially among early photosynthetic life forms. Experiments with modern green algae, the Volvox lineage, illustrate evolutionary steps mirrored in the fossil record, from single cells, Chlamydomonas, to colonies of identical cells, Gonium, to differentiated spheroids, Volvox. Each stage adds a single genetic innovation, adhesion, polarity, cell cycle coordination, showing that complexity can arise gradually through co-option, not sudden design.
Oxygen and nutrients, catalysts of complexity.
During the middle part of the Proterozoic eon, Earth's environment placed strict limits on how life could evolve. Oxygen levels in the atmosphere and oceans stayed low for hundreds of millions of years, but every so often, around 1.4, 1.1, and 0.8 billion years ago, brief whiffs of oxygen appeared. Each of these oxygen spikes coincided with periods when complex eukaryotic life began to diversify. Scientists think these bursts of oxygen followed times of continental uplift and increased rock weathering, which released nutrients like phosphorus and iron into the oceans. These nutrients boosted biological activity, creating short-lived zones of higher oxygen that helped drive evolutionary progress. Chemical clues preserved in ancient marine rocks reveal how oxygen levels in Earth's oceans gradually increased over time. Between 1.6 and 1.0 billion years ago, the amount of the metal molybdenum in ocean sediments rose from very low levels to much higher ones, showing that deep waters were becoming periodically oxygenated. At the same time, variations in chromium isotopes point to bursts of oxygen-driven weathering on land. These chemical spikes line up with major biological milestones: the rise of red and green algae, the growing diversity of acritarch microfossils, and the first appearance of microscopic remains rich in sterols, compounds found in the cell membranes of complex life. Phosphorus, a key nutrient for all living things, played a major role in controlling how much life the oceans could support. As continents eroded and rocks rich in the mineral apatite broke down, phosphorus was released into rivers and seas, fueling the growth of algae. Scientists call this process the phosphate feedback. As more oxygen spread through soils and surface waters, more phosphorus became available, which in turn stimulated algal blooms. When these algae died and were buried, they locked away carbon and allowed even more oxygen to build up in the atmosphere. This positive feedback loop helped push Earth's environment toward greater biological complexity.
Early eukaryotic ecosystems.
By 1 billion years ago, shallow marine platforms hosted complex microbial mats composed of cyanobacteria, algae, and heterotrophic protists. Stromatolites became less common, replaced by layered microbial algal communities that stabilized sediments and formed the first benthic ecosystems. Planktonic algae filled the photic zone, driving new carbon and nutrient cycles. After about 1.1 billion years ago, the chemical makeup of carbon in ancient rocks began to vary much more than before, showing that life was becoming more active and that more organic material was being buried in sediments. Black shales from this time contain chemical fossils called steranes, which come from red and green algae, clear evidence that photosynthetic eukaryotes were becoming more diverse. At the same time, changes in the ratios of nitrogen isotopes reveal that Earth had developed a fully functioning nitrogen cycle, with processes like nitrification and denitrification that depend on oxygen. Together, these clues show that by this point, both the biosphere and atmosphere had grown far more dynamic and oxygen-rich than ever before. Microscopic predation emerged. Fossils of Melanocerillium and Paleomastus show openings interpreted as feeding structures. The evolution of phagocytosis among protists fundamentally changed nutrient flow, recycling organic matter and linking food webs for the first time. Ecology had begun in earnest.
The emergence of differentiation.
Once oxygen, energy, and genetic diversity converged, some lineages experimented with cellular specialization. Colonial organisms divided labor between somatic and reproductive cells, paving the way for tissues. Communication evolved through chemical signaling, precursors to hormones and neurotransmitters. The cell had learned cooperation. Groups of genes that control how cells develop and specialize, known as homobox and MADS-box genes, first appeared around 1 billion years ago. These genes act like master switches, turning other genes on and off to guide the formation of different cell types and structures. The earliest evidence of their use comes from red algae, where they help regulate the production of spores and gametes for reproduction. These genetic innovations arose through a mix of gene duplication and horizontal gene transfer, the sharing of DNA between different organisms. Once sexual reproduction evolved, these new genes could spread and combine in new ways, driving the emergence of greater complexity in early eukaryotic life. Depth cutaway. In rocks from the Torridonian group in Scotland, dating to about 950 million years ago, scientists have found fossils of multicellular algae up to 300 micrometers wide. These tiny organisms show clear signs of cell walls and organized patterns of cell division, evidence that their growth was carefully coordinated rather than random. Preserved in sediments from ancient river deltas and lakes, these fossils record a key evolutionary moment, the shift from simple microbial mats to the earliest forms of tissue, representing life's first steps toward organized, multicellular structure.
Transition summary.
By the close of the Mesoproterozoic, life had reinvented itself once again. Sex introduced shuffling and innovation. Multicellularity introduced structure and cooperation. Oceans once dominated by single cells now contained colonies and filaments, photosynthetic forests of microscopic complexity. The stage was set for the next transformation, the Ediacaran world, when multicellular life would fill the seas with form.
Part 4. The Ediacaran Dawn and Prelude to Complex Life. The Ediacaran Threshold.
By 900 million years ago, Earth's biosphere was ready for another leap. The long stability of the Mesoproterozoic began to break. Supercontinents drifted, climates fluctuated, and oxygen levels crept upward. In this changing world, the quiet lineage of eukaryotes, once confined to microscopic realms, began to expand into visible forms. Sheets, filaments, and disks of soft-bodied organisms colonized the seafloor. Their traces mark the dawn of macroscopic life, the Ediacaran period, bridging the gap between the microbial Earth and the animal world to come. The Ediacaran period, which lasted from about 635 to 541 million years ago, began after the dramatic global ice ages of the Cryogenian, when much of Earth had been frozen over. As the planet thawed, oxygen levels in the atmosphere rose sharply, possibly reaching up to 1/5 of what we have today. At the same time, melting glacier released nutrient-rich runoff from the continents into the oceans, fueling an explosion of biological activity. Chemical evidence from carbon isotopes shows that organic matter was being produced and buried at high rates, meaning the carbon cycle had become much more dynamic. Together, these changes created the oxygen-rich, fertile conditions that allowed complex multicellular life to flourish for the first time.
Continental motions and climate instability.
During the Neoproterozoic era, the giant supercontinent Rodinia slowly came together and then broke apart again, reshaping Earth's surface. As the continents drifted, new coastlines and shallow seas formed, changing ocean currents and climate patterns. The breakup also exposed vast areas of rock to weathering, which pulled carbon dioxide out of the atmosphere and may have caused two extreme global ice ages: the Sturtian, around 720 million years ago, and the Marinoan, around 635 million years ago. These snowball Earth events nearly froze the planet solid, posing huge challenges for life. Yet when the ice finally melted, the newly exposed rocks released nutrients into the oceans, sparking a surge in biological growth that helped set the stage for the rise of complex life. Chemical clues preserved in ancient rocks reveal how dramatically Earth's environment shifted during and after the great ice ages. Layers of carbonate rock that formed directly above glacial deposits show very low carbon isotope values, a sign that huge amounts of buried organic material were rapidly oxidized once the planet began to warm. At the same time, changes in strontium isotopes point to a period of intense erosion and weathering on the continents as the ice melted. These chemical signals coincide with a surge in sterane molecules, which come from algae, showing that photosynthetic life quickly rebounded in the newly fertile oceans after each global thaw. Computer models of Earth's ancient climate show that it took an enormous buildup of carbon dioxide in the atmosphere, more than 300 times higher than today's pre-industrial levels, to melt the planet out of its global ice age. When the ice finally gave way, this intense greenhouse effect caused surface temperatures to soar above 40 degrees Celsius for thousands of years. The sudden heat put great stress on surviving ecosystems, but it also drove a burst of chemical activity and rapid environmental change, creating new opportunities for evolution and the emergence of more complex life.
The second oxygenation.
After the planet thawed from the Marinoan Ice Age about 635 million years ago, Earth's environment underwent another major transformation known as the Neoproterozoic oxygenation event. During this time, oxygen levels rose higher than ever before, finally reaching amounts that could support large, energy-demanding organisms. This surge in oxygen changed the chemistry of the oceans, making them more hospitable to complex life. As a result, the first multicellular animals began to appear, along with the earliest interconnected food webs, marking the beginning of a new and dynamic stage in Earth's biological history. Chemical evidence from ancient rocks shows that by about 580 to 560 million years ago, Earth's deep oceans had finally become rich in oxygen. Measurements of iron and molybdenum isotopes in dark shale layers reveal that oxygen had spread throughout the seafloor, while chromium isotopes indicate that oxygen-driven weathering was also happening widely on land. Around the same time, large phosphate-rich deposits, such as those found in the Dushantuo Formation in China, began to form, showing that both oxygen levels and biological productivity had increased. The oceans also became richer in sulfate, reaching about 10% of modern levels, which supported new types of sulfur-based metabolisms and helped early organisms begin to build shells and skeletons. This rise in oxygen was closely linked to changes in life itself. As large photosynthetic algae became more common, they absorbed carbon dioxide and buried more organic carbon in ocean sediments, which allowed oxygen levels to rise. Higher oxygen, in turn, made it possible for larger organisms to evolve, ones that needed more energy and oxygen to survive. This created a powerful feedback loop. More life produced more oxygen, and more oxygen allowed life to become more energetic and complex, setting off one of the most important evolutionary chain reactions in Earth's history.
Eukaryotic diversification.
With both oxygen and energy now plentiful, eukaryotic life began to diversify at an astonishing pace. Red, green, and brown algae spread across the oceans, forming vast underwater forests, while fungus-like organisms began to grow within sediments on the seafloor. For the first time, predatory single-celled organisms appeared, hunting and consuming other microbes. Fossils from between 1 billion and 600 million years ago show an incredible variety of acritarchs, tiny, intricately patterned microfossils that represent the plankton of that era. Their growing size, complexity, and global abundance mark a major ecological turning point, the worldwide expansion of complex eukaryotic life. The variety of acritarchs, tiny fossilized cells thought to represent ancient plankton, expanded dramatically between 900 and 700 million years ago. Studies of hundreds of rock formations around the world show that what was once a time of low diversity, with fewer than a dozen types in each sample, gave way to a period bursting with over 100 distinct forms. Detailed measurements reveal that some of these cells grew up to 0.5 millimeter across and developed thick, multilayered walls, features that would have required far more energy and oxygen to sustain. This explosion in size and complexity reflects how rapidly life was responding to the newly enriched, oxygenated oceans of the Neoproterozoic world. Chemical traces known as biomarkers provide a hidden record of life that existed long before animals left visible fossils. Compounds such as 24-isopropylcholestane, made by sponges, and dinosterane, produced by dinoflagellates, a type of advanced plankton, have been found in rocks dating from 635 to 600 million years ago. These molecules act as molecular fossils, revealing that early animals and complex single-celled organisms were already living in the oceans well before they appeared in the fossil record. They mark the dawn of visible life, the first hints of the larger creatures soon to come.
Experiments inform the Ediacaran organisms.
By about 575 million years ago, the fossil record reveals the first communities of large, soft-bodied organisms, the Ediacaran biota. Found in the sandstones and siltstones of Newfoundland, Namibia, and Australia, these organisms show quilted, frond-like, or radial patterns unlike any modern species. Some resembled ferns anchored to the seafloor, Charnia, fractofusus, others with discs or pouches embedded in microbial mats. They may have absorbed nutrients directly through their surfaces, lacking mouths, guts, or skeletons. Some of the most beautifully preserved fossils from the Ediacaran period come from the Mistaken Point Formation in Newfoundland, dating to about 565 million years ago, and the Ediacara Hills in South Australia, about 555 million years old. These sites are precisely dated using uranium lead measurements from volcanic ash layers found between the fossil beds. The fossil surfaces reveal entire communities of organisms, with individuals aligned in similar directions and showing a range of sizes. Patterns that suggest these early life forms interacted with one another and may even have reproduced sexually. Chemical evidence from the rocks where these fossils are found shows that the oceans at the time were rich in oxygen. Measurements of iron and molybdenum confirm that even the deepest waters had become fully oxygenated. A large shift in carbon isotope values, known as the Shuram anomaly, suggests that huge amounts of buried organic matter were suddenly oxidized, which briefly lowered oxygen levels but also released nutrients into the oceans. These environmental ups and downs may have acted as a spark for evolution, driving the rapid diversification of multicellular life that defines the late Ediacaran world.
The Ediacaran ecosystem.
Ediacaran seafloors were microbial landscapes, thick mats of cyanobacteria and algae-bound sediments, forming stable surfaces on which larger organisms could attach. Within these mats, chemical gradients supported diverse microbial communities, sulfur reducers below, oxygenic phototrophs above. The new macroscopic organisms exploited this layered ecosystem, grazing, absorbing, or symbiotically associating with microbes. Trace fossils, grooves, burrows, and feeding trails first appear in the late Ediacaran. Simple and branched tubes, planolites, triptychnes, mark the earliest evidence of locomotion and behavior. They record oxygenated substrates supporting active metabolism. These tracks prefigure the rise of bilaterian animals in the Cambrian. Chemical fossils preserved in Ediacaran age rocks show that by this time, Earth's ecosystems had become both balanced and complex. Molecules called steranes, from eukaryotes, and hopanes, from bacteria, appear in roughly equal amounts, suggesting that both kinds of life were thriving together in stable, energy-rich environments. At the same time, the ratios of nitrogen isotopes indicate that the planet had developed a fully functioning nitrogen cycle, where oxygen played a key role in recycling nutrients. This system could support multiple levels of the food chain, marking an important step toward the modern, interconnected ecosystems we know today.
The genetic foundations of animal life.
Genetic studies show that the blueprint for building animals existed long before the famous Cambrian explosion. Many of the key genes that control how cells stick together, communicate, and specialize, such as integrins, cadheins, and the WNT, notch, and hedgehog pathways, first evolved in single-celled organisms related to animals, including choanoflagellates and philisterians. These early microbes used the same genes for simpler purposes, like attaching to surfaces or sensing their surroundings. Later, evolution repurposed this ancient genetic toolkit to create tissues, organs, and body plans, laying the groundwork for the first multicellular animals. Genetic comparisons suggest that animals split from their single-celled ancestors sometime between 800 and 750 million years ago. Chemical fossils called 24-isopropyl cholestanes, produced by early sponges, appear in rocks from the same period, supporting this timeline. By then, oxygen levels had reached about 10 to 20% of modern concentrations, enough to fuel the energy needs of multicellular organisms. The fossils of the Ediacaran period, though often mysterious in form, capture the first flourishing of these new kinds of life, creatures made possible by both rising oxygen and an increasingly sophisticated genetic toolkit. Experiments on modern sponges, such as amphimedon, show that even with a relatively small set of about 18,000 genes, they can still form distinct tissues and regulate a simple immune system. This suggests that the basic genetic machinery needed to build and maintain a multicellular body is surprisingly minimal. These ancestral genetic systems probably first appeared in Ediacaran age organisms, representing an important evolutionary bridge between simple microbial colonies and the first true animals.
The end of the Ediacaran.
Around 541 million years ago, the Ediacaran world vanished, changing ocean chemistry, rising predation, and ecosystem restructuring erased its softly bodied organisms. Yet their legacy endured. They had proven that complex multicellular life was possible. Within a few million years, the Cambrian explosion would fill the seas with shells, skeletons, and motion, the visible world of animals. Around 541 million years ago, Earth underwent a major environmental and biological transformation. Many of the distinctive Ediacaran organisms disappeared, while new trace fossils show that animals capable of burrowing and moving through sediments had emerged. At the same time, the chemistry of the oceans changed dramatically. Sulfate levels doubled, and rising calcium carbonate made it easier for creatures to develop shells and skeletons. Shifts in carbon and sulfur isotopes reveal that the planet's carbon-sulfur cycle was being reorganized, likely due to expanding zones in the ocean with little oxygen. Although these changes disrupted existing ecosystems, they also set the stage for the rise of energetic, complex animals that would soon dominate the Cambrian world.
Conclusion: the hidden revolution.
The Proterozoic Eon, long dismissed as uneventful, was in truth the crucible of complexity. In its billion years of quiet, life discovered partnership, sex, and cooperation. Cells merged to form organelles, individuals joined to form tissues, and genes evolved into networks capable of building bodies. By the close of the Ediacaran, the blueprints of every later organism already existed in microscopic form. The next chapter, the Cambrian explosion, would make this hidden revolution visible.
Episode 8: The Cambrian Explosion, Burst of Animal Diversity in Oceans. Part 1: The World Before the Explosion. Introduction: a planet poised for change.
541 million years ago, Earth stood at a turning point. For billions of years, the planet had teemed only with microbes, algae, and soft-bodied organisms whose delicate forms left little trace in stone. Then, over an interval short by geological standards, the oceans filled with shells, limbs, eyes, and jaws. Almost every major animal design that would ever exist appeared for the first time. To later observers, this transformation would seem sudden, a literal explosion of life, but it was built upon long preparation. The Cambrian world inherited the chemical and biological legacies of the Ediacaran: oxygenated seas, nutrient-rich shelves, and genetic toolkits already primed for complexity. The Cambrian explosion unfolded between 541 and 515 million years ago, an interval of roughly 25 million years in which taxonomic diversity increased tenfold and morphological disparity approached modern levels. The event marks the formal beginning of the Phanerozoic Eon, the time of visible life. Exceptional fossil sites across every continent preserve both soft and hard tissues, allowing this period to be reconstructed with unusual precision.
The Earth at the turn of the Eon.
The early Cambrian Earth was warm and restless. The supercontinent Rodinia had fragmented into smaller landmasses: Laurentia, Baltica, Siberia, and Gondwana, surrounded by broad, shallow seas. These continental shelves were fertile nurseries, bathed in sunlight and stirred by tides that kept nutrients in motion. Atmospheric oxygen, rising since the late Proterozoic, hovered near modern surface values. Calcium and phosphate, washed from weathering rocks, enriched the oceans and tipped their chemistry toward mineral saturation. At the start of the Cambrian, the chemistry of ancient rocks shows a major shift in Earth's carbon cycle. More organic carbon was being buried in sediments, which in turn meant more oxygen was being produced and left in the atmosphere and oceans. Other chemical clues, like unusually high levels of molybdenum, indicate that even the deep ocean was starting to fill with oxygen. Measurements of iron minerals back this up. They show that oxygen-rich conditions were spreading widely. Together, these changes created an environment where animals that needed lots of energy, and therefore lots of oxygen, could finally flourish. Reefal carbonates built by archaeocyathid sponges appear by 525 million years. These organisms required both clear, oxygenated water and stable carbonate saturation. Their framework reefs, found today in Siberia and Antarctica, are among the earliest large ecosystems constructed by animals and provide tangible evidence of a newly oxygen-rich biosphere.
The Cambrian substrate revolution.
Before animals learned to burrow, the seafloor was bound by dense microbial mats. Ediacaran life clung to or rested upon these surfaces, but rarely disturbed them. At the dawn of the Cambrian, small worm-like creatures began to dig. Their movement mixed the sediment, carried oxygen deeper into the mud, and destroyed the old mat communities. This behavioral shift, the Cambrian substrate revolution, transformed marine ecology forever. The appearance of the sinuous trace fossil Treptichnus pedum defines the official base of the Cambrian in the global stratigraphic scale. These complex, branching burrows record three-dimensional exploration of sediment by bilaterian animals capable of coordinated movement and sensory feedback. Sediment redox profiles changed dramatically. Oxygen penetration increased from less than a millimeter in the Ediacaran to several centimeters by 530 million years, expanding habitable microenvironments and creating niches for detritivores and predators alike. This new churning of the seabed altered global geochemistry. Organic matter buried in anoxic muds was now exposed to oxygen, accelerating decay and nutrient recycling. Phosphate regenerated into the water column, fueling algal blooms, while carbon dioxide release stabilized the carbon cycle. For the first time, animal behavior itself became a planetary force.
The invisible preparation.
Although the Cambrian explosion seems abrupt, its genetic and biochemical groundwork had been laid long before. During the late Proterozoic, multicellular organisms had already evolved the fundamental developmental machinery: cell adhesion, signaling pathways, and pattern gene expression that would later shape animals. What changed was the environment's ability to support them. Rising oxygen allowed these dormant potentials to express themselves as anatomy. Molecular clock analyses of conserved genes place the divergence of major animal lineages, sponges, cnidarians, and bilaterians, between 650 and 600 million years ago, well before their fossil appearance. The explosion therefore represents not the birth of animals but their ecological release, when physical and chemical thresholds finally permitted large, mobile, energy-demanding forms to persist. In parallel, oceanic calcium concentrations climbed to roughly 25 millimoles per liter, double the Proterozoic norm. This shift destabilized organic membranes and favored biomineralization as a regulatory mechanism for excess ions. What began as internal detoxification would soon produce shells, skeletons, and reefs.
The first skeletal organisms.
The earliest tangible relics of this new world are the small shelly fossils scattered through lower Cambrian strata, tiny tubes, scales, and spicules composed of calcium carbonate or phosphate. They are the first widespread evidence of animals constructing durable skeletons. Some belong to primitive mollusks and worms. Others, like Cloudina and Anaborites, built nested cones that foreshadowed corals and annelids. Thin-section microscopy reveals growth lines and microgranular fabrics identical to modern biologically controlled minerals, proving that these were true biominerals rather than chemical precipitates. Oxygen isotope ratios within the shells yield seawater temperatures of 30 to 35 degrees Celsius, consistent with warm, equatorial shallow seas that promoted rapid carbonate deposition. The ecological consequences were immediate. Hard parts provided protection and new surfaces for colonization, launching an evolutionary arms race between predators and prey. Mineral skeletons also increased sediment stability and carbonate production, feeding back into ocean chemistry and further stimulating reef growth.
A world transformed.
By 530 million years ago, the quiet microbial seas of the Proterozoic had become vibrant ecosystems filled with movement and form. Burrowers re-engineered the seafloor, shelled organisms built reefs, and oxygen-rich waters sustained the first food webs of macroscopic animals. The stage was set for the flowering of the Cambrian fauna, the appearance of eyes, claws, and intricate bodies recorded in places like Chengjiang and the Burgess Shale.
Part 2. The explosion unfolds. Eyes, predators, and the Burgess Shale world. The opening of the Cambrian seas.
Within only a few million years of the first shelly fragments, the marine world became unrecognizable. Burrowing and skeletons had re-engineered the substrate. Now movement and predation reshaped the water column itself. Organisms that once drifted passively learned to crawl, swim, and hunt. The Cambrian oceans turned dynamic. Clouds of plankton drifted above reefs and sandbars. Worm-like creatures plowed through the mud, and new predators patrolled the depths. For the first time in Earth's history, life possessed eyes, armor, and strategy. Radiometric dating places the main pulse of diversification between 530 and 515 million years ago. Within this span, the fossil record of every major marine phylum appears: arthropods, mollusks, annelids, brachiopods, echinoderms, and chordates. Measured in evolutionary tempo, morphological disparity increased faster during this interval than at any later time in the history of animals.
The birth of predation.
Predation, more than any other ecological force, drove the Cambrian transformation. Once some animals began to consume others, every lineage faced new selective pressures. Defensive skeletons, rapid motion, sensory organs, and complex behaviors evolved in quick succession. The simple microbial mats of the Ediacaran could not have withstood such an assault. They vanished, replaced by living sediments filled with predators and prey. The appearance of boring traces in early Cambrian shells, tiny drill holes less than a millimeter across, marks the first direct evidence of predation. The shells of Cloudina from Namibia and Oman show circular perforations made by viewer-like mouthparts. These fossils predate the main explosion by nearly 10 million years, revealing that predation began locally before spreading globally. Experiments with modern marine ecosystems show that a single introduction of a new predator can increase prey diversity twofold through the creation of refuges and adaptive specialization. In the Cambrian, this ecological feedback occurred across entire oceans, amplifying diversity on a planetary scale.
The evolution of vision.
To hunt or flee required perception. The earliest eyes appear among Cambrian arthropods, compound organs composed of hundreds of lenses capable of detecting movement and shape. Trilobites, which dominated the seas for the next 270 million years, carried calcite-reinforced eyes whose optical precision rivaled modern insects. With sight came the birth of complex behavior, pursuit, evasion, and recognition. The earliest known hard, mineral-based eyes appear in a trilobite called Olanellus, about 521 million years ago. Each eye lens was made from a single, perfectly aligned crystal of calcite, a natural mineral, arranged so that light could pass through cleanly. This is one of the earliest examples of evolution turning a rock-like material into a working optical device. Similar eye designs are found in the fierce predator Anomalocaris from the Chengjiang and Burgess shale fossils. Its stalked eyes held up to 16,000 tiny lenses, giving it vision on the level of a modern dragonfly. The emergence of vision altered evolutionary dynamics. Predators could target moving shapes. Prey could detect approaching danger. This visual arms race explains the simultaneous proliferation of spines, camouflage, and rapid locomotion seen in Cambrian fossils. Evolution accelerated through the feedback of seeing and being seen.
The rise of the arthropods.
Among the most successful Cambrian animals were the arthropods, creatures encased in jointed exoskeletons that combined protection with mobility. From their ranks descended trilobites, crustaceans, and insects, the dominant forms of later eras. In the Cambrian, they occupied every niche: swimmers, burrowers, scavengers, and hunters. The Burgess Shale preserves incredibly detailed fossils of early arthropods like Marella, Yohoia, and Wapsha. Their well-developed gills, complex eyes, and jointed legs show that the basic arthropod body design was already advanced by 508 million years ago. Chemical studies of the fossils even reveal traces of chitin and proteins, the same materials found in the hard exoskeletons of modern insects and crustaceans. DNA-based molecular clock studies place the split between major arthropod groups, such as crustaceans and spiders, at around 540 to 530 million years ago, which fits perfectly with what the fossils show. Trilobites first appeared about 521 million years ago and quickly evolved into hundreds of different species. Because they spread all over the world and changed rapidly through time, geologists use them as index fossils to help date ancient rocks. Huge numbers of their shed exoskeletons have been preserved, showing that trilobites regularly molted as they grew. The patterns of these molts suggest they may have had complex life cycles and even gathered in groups to shed their shells, possibly timed with the tides or the phases of the moon.
The Burgess Shale and Chengjiang faunas.
The fullest view of the Cambrian world comes from two extraordinary fossil sites: the Chengjiang biota of Yunnan, China, dated 518 million years ago, and the Burgess Shale of British Columbia, 508 million years ago. Both preserve soft tissues in astonishing detail: eyes, gills, intestines, even nerve cords, allowing reconstruction of entire ecosystems. The Chengjiang Shale contains over 250 species representing at least 16 animal phyla. Among them are the earliest known chordates, Hycoichthyes, and Mylocunmingia, which possess notochords and segmented muscles, the first vertebrate relatives. Pyritized gut tracts reveal predatory diets, while delicate fronds of early algae show the base of the food chain. The Burgess Shale, deposited in a deep shelf basin below a carbonate reef, owes its preservation to rapid burial by underwater mudslides in oxygen-poor waters. Chemical analyses show that fine aluminosilicate films replaced soft tissues before decay, capturing even cellular detail. Species such as Opobinia, with 5 eyes and a grasping trunk, or Anomalocaris, a meter-long apex predator, illustrate how experimental and unbounded Cambrian design could be.
The dawn of the chordates.
Amid the riot of arthropods and worms appeared a lineage that would eventually produce fish, amphibians, reptiles, and mammals. The earliest chordates were small, translucent swimmers with stiffened dorsal rods and gill slits for filter feeding. Their emergence marks the beginning of vertebrate history. Hycoichthyes erkeicunensis from Chengjiang, only 2.5 centimeters long, shows segmented myomeres, a notochord, and paired eyes, definitive chordate characters. Geochemical mapping of phosphorus confirms that its internal skeleton was non-mineralized, composed of collagen rather than bone. Slightly younger forms such as Pichia gracilans from the Burgess Shale display more advanced musculature, indicating early experimentation with undulatory swimming. The origin of the vertebrate head, a concentration of sensory organs and a central nervous system, is traced through these fossils. Gene expression patterns in modern fish embryos mirror the segmented structures seen in Hycoichthyes, linking developmental genetics directly to Cambrian anatomy.
The structure of Cambrian ecosystems.
By the height of the explosion, the sea supported complex food webs. Photosynthetic plankton formed the base, grazed by filter-feeding sponges, brachiopods, and early mollusks. Worms and trilobites scavenged detritus, while cephalopod-like predators and anomalocaridids ruled above. For the first time, energy flowed through multiple trophic levels, recycling nutrients and supporting large populations. Chemical signatures in Cambrian rocks show that the food chain of the time was already organized much like today's oceans. The nitrogen values increased steadily from one step of the food web to the next, revealing a clear hierarchy of producers, consumers, and top predators. Studies of the Burgess Shale also show layered communities, animals burrowing in the mud, others living on the seafloor, and swimmers above them. This kind of structured, multi-level ecosystem had never existed before in Earth's history.
Transition summary.
By the close of the early Cambrian, life had achieved complexity and interaction undreamed of in the Proterozoic seas. The invention of sight and predation created evolutionary feedbacks that filled the oceans with diversity. Hard skeletons fossilized this abundance, ensuring that from this point onward, the history of life would be written indelibly in stone.
Part 3. The architecture of animal life. The blueprint of bodies.
The Cambrian seas were filled with new shapes: coiled shells, jointed limbs, segmented trunks, and radiating tentacles. Beneath this astonishing variety lay a shared biological grammar, a system of genetic instructions capable of building eyes, wings, and hearts from a single ancestral script. The discovery of this hidden unity has transformed our understanding of the explosion. What appeared as a riot of unrelated forms was, in truth, the unfolding of a deep developmental architecture already in place before the Cambrian dawn. The key to this architecture is the Hox gene cluster, a family of genes that organize the body along its head-to-tail axis. Each Hox gene encodes a transcription factor that activates specific developmental pathways in defined regions. The sequence of these genes on the chromosome mirrors the order of their expression in the embryo, an arrangement known as collinearity. Comparative genomics shows that all bilaterian animals, arthropods, worms, mollusks, and vertebrates share the same fundamental Hox pattern. The duplication and diversification of these genes between 700 and 600 million years ago created the molecular potential for every later body plan. Fossils of early animals like Pichia and the first segmented worms show that bodies with repeated segments had already evolved. Chemical traces from the same time indicate that these animals were becoming more active and energetic. Oxygen measurements in Cambrian rocks also point to warmer oceans and busier ecosystems. Together, these clues show how new genetic abilities and rising environmental energy worked hand in hand to drive early animal evolution.
Skeletons. Chemistry turned to architecture.
Mineral skeletons, first seen as tiny shelly fragments, soon evolved into elaborate frameworks. Different groups adopted distinct strategies. Some used calcium carbonate in its calcite or aragonite forms, others secreted calcium phosphate or silica. These biominerals not only protected the organism but also acted as scaffolds for muscle attachment and mechanical leverage, allowing larger and more powerful movement. The widespread appearance of hard shells and skeletons began when the oceans became rich enough in calcium carbonate for minerals to form easily. Models suggest this happened because more calcium and dissolved carbon washed into the sea as the ancient supercontinent Rodinia broke apart. Early animals with shells, such as anaborites and Hyalothellus, built them from a form of calcite rich in magnesium. As ocean chemistry shifted back and forth over time, later organisms switched to using aragonite instead. These changes reflect how ocean conditions shaped the kinds of skeletons early animals could make. Phosphatic skeletons, such as those of early brachiopods and small shelly fossils like Microna, indicate local upwelling of phosphate-rich waters. Silicon-based structures, including sponge spicules, record distinct oceanic provinces with high-dissolved silica. These regional differences in seawater chemistry produce the patchwork of biomineral types visible in Cambrian sediments. The formation of skeletal reefs, archaeocyathid sponge frameworks and calcimicro buildups represents the first large-scale biological modification of Earth's crust. Their construction trapped sediment, altered carbonate saturation, and created microhabitats for countless smaller organisms. Reef growth rates, measured from vertical accretion in fossil sections, reach 20 meters per million years, comparable to modern coral systems.
The developmental explosion.
Behind the morphological bloom lay a genomic one. Analyses of gene families across modern animals reveal a burst of duplication events around the Proterozoic Cambrian boundary. These duplications created redundant copies free to specialize, giving rise to novel cell types, signaling molecules, and tissue structures. Multicellularity had provided the framework. Gene duplication filled it with diversity. Molecular clock studies of transcription factor superfamilies, including the homobox, basic helix, loop helix, and zinc finger groups, show peaks in duplication between 650 and 550 million years ago. This expansion correlates with the fossil evidence for increasing cell type complexity, from simple epithelia in Ediacaran organisms to true organs in Cambrian taxa. The emergence of immune genes, structural proteins, collagen, elastin, and neural channels all date from the same interval, forming the biological infrastructure of animal physiology. The neurobiological revolution of the Cambrian is reflected in trace fossils showing directed movement and predatory pursuit. Neuron-rich nerve cords are preserved as carbonaceous films in Fuxian Huia from Chengjiang, demonstrating that arthropods already possessed centralized brains with optic lobes. These fossils link neural complexity directly to ecological innovation.
Tiered ecosystems, life builds the landscape.
The Cambrian seafloor was no longer a flat microbial plane. For the first time, life occupied vertical space. Burrowers lived beneath the sediment, sessile filter feeders and grazers anchored to the surface. Swimmers and predators filled the water column above. This ecological tiering established the structure of marine communities that persists to this day. Fossil bedding plane analyses from Burgess Shale localities show tiered assemblages with three dominant levels. In faunal burrowers at 0 to 5 centimeters depth, benthic organisms fixed to the surface, and nectanic swimmers within the water column. Energy transfer between these tiers produced efficient recycling systems, reducing the accumulation of organic detritus. Bioturbation indices rise from 0 to 4 across the early Cambrian, quantifying this expansion of ecological depth. Changes in the global carbon cycle show how life was reorganizing the planet during the Cambrian. The chemistry of Cambrian limestone rocks rises and falls in a steady rhythm, reflecting repeated periods when large amounts of organic material were buried and later exposed again. These natural cycles helped keep the level of carbon dioxide in the atmosphere fairly stable, high enough to maintain a warm climate, but not so high that the planet overheated. In this way, early ecosystems began to help regulate Earth's temperature.
Cutaway, the geometry of evolutionary space.
Paleontologists describe the Cambrian expansion not merely as a rise in diversity but as a filling of morphospace, the abstract volume of all possible anatomical designs. Early in the period, new body plans appeared in almost every available region of this conceptual space: segmented worms, jointed limbs, radial forms, shells, spines, and soft-bodied swimmers. Later evolution would elaborate within these existing frameworks but rarely invent new ones. Quantitative analyses of morphological disparity using principal coordinate data from Cambrian and Ordovician faunas show that by 515 million years ago, total morphospace occupation exceeded 80% of the range achieved by the present day. This pattern indicates that the explosion's essence lay not in species count, but in the origination of fundamental body architectures, a single burst of innovation that defined the limits of animal design.
Stabilization after the burst.
By the Middle Cambrian, around 510 million years ago, the frenetic pace of innovation slowed. Ecosystems stabilized. Extinction and diversification reached equilibrium. Trilobites, brachiopods, mollusks, and echinoderms settled into enduring lineages. The evolutionary frontier shifted from invention to refinement. Natural selection honed the efficient forms that would dominate for hundreds of millions of years. Fossil assemblages from the Drumian stage, tilde 504 million years ago, show community-level stability lasting millions of years, with turnover rates below 0.2% per 100,000 years, the earliest known ecological equilibrium. This marks the transition from the explosive to the consolidating phase of the Phanerozoic biosphere.
Transition summary.
The Cambrian world was not a chaos of monsters but a structured experiment in possibility. Genetic architecture, chemical opportunity, and ecological interaction converged to produce the living geometry of animals. The body plans forged in these seas, segmented limbs, exoskeletons, gills, and eyes, would persist, with modification, into every future epoch. In the next chapter of Earth's history, this new biological complexity would stabilize and expand beyond the shallow shelves, giving rise to enduring ecosystems and the first great extinctions.
Part 4. Stabilization and Legacy. From Cambrian Seas to the Ordovician World. After the storm.
By roughly 505 million years ago, the explosive burst of new body forms began to slow. The once experimental Cambrian biosphere matured into stable ecosystems with defined trophic structures. Life had colonized every available marine niche, burrowing sediments, reef surfaces, pelagic waters, and the pattern of evolutionary radiation followed by stabilization had become established for the first time. The chaos of novelty gave way to the order of persistence. From this balance emerged the foundations of the modern marine world. Fossils from the Middle to Late Cambrian show an important shift in how life was evolving. The number of species kept increasing, but the variety of body shapes stopped expanding as quickly. Most of the major designs had already appeared. Measurements of these fossils show that by about 510 million years ago, animals were already filling more than 80% of the basic design space we see today. At the same time, extinctions became far less frequent than they had been earlier in the Cambrian. This drop in turnover marks the beginning of a more stable and balanced set of ecosystems.
The persistence of the trilobite world.
Among the enduring victors of this new equilibrium were the trilobites. Their chitinous exoskeletons, compound eyes, and flexible segments proved adaptable to nearly every marine habitat. Trilobite evolution through the later Cambrian reveals both conservatism and innovation. Conservative in their overall body plan, yet endlessly inventive in form and ornament. They became the visual emblem of the Paleozoic seas. Over 20,000 trilobite species are known, spanning 270 million years of history. Cambrian trilobites show rapid provincial diversification: Olanellids in Laurentia, redlichids in Gondwana, paradoxidids in Baltica, indicating the emergence of distinct continental faunal realms. Growth lines in their eyes and exoskeletons record tidal and seasonal rhythms, demonstrating not only biological sophistication but also that Earth's day length had already stabilized near 21.5 hours. Trilobites' molting behavior created mass accumulations of shed exoskeletons preserved as fossil beds. Such deposits serve as ancient census points of population dynamics. Geochemical analyses of associated shales reveal periodic anoxia in deeper settings, showing that even amid overall oxygenated seas, local redox gradients continued to shape communities.
Reefs, currents, and carbonate worlds.
Cambrian reefs, initially built by archaeocyaths and microbial consortia, expanded into immense carbonate platforms. Their framework structures not only provided habitats but also influenced ocean circulation and chemistry. By trapping sediments and modifying local hydrodynamics, They established feedbacks between biology and geology that would echo throughout the Phanerozoic. Archaeocyath reefs, built by ancient sponge-like organisms, were thriving between about 525 and 515 million years ago, but then they suddenly declined, probably because the oceans cooled and their chemistry changed. Microscopic studies of these reefs show that they were held together by thin layers of helpful microbes that grew alongside the sponges. Chemical patterns inside the reef rocks rise and fall in regular cycles, showing times when large amounts of carbon were buried by reef growth. Their collapse happened just as the ocean shifted from favoring one type of mineral for shells and skeletons to another, a change caused by shifts in seafloor activity deep within the Earth. The huge shallow water platforms of the Cambrian, covering more than 30 million square kilometers, played a major role in removing carbon dioxide from the atmosphere. As these vast seafloor areas slowly broke down and reacted with seawater, they helped lock carbon into rocks. Layered sediments built up steadily, showing that living organisms were constantly producing carbonate material that formed new seafloor. Over time, these enormous platforms hardened into limestone, which later became the core of many mountain ranges. In a very real sense, today's mountains are built on the remains of Cambrian reefs.
Extinctions and recoveries.
Stability was never absolute. During the Cambrian, life went through at least 3 major extinction events. Each one wiped out many species and opened up space for new kinds of organisms to evolve. The biggest of these extinctions happened around 485 million years ago, right at the transition into the Ordovician period. It wiped out many families of trilobites, the last of the archaeocyaths, and several early groups of echinoderms. But within just a few million years, the surviving species spread out and evolved into a wide variety of new forms. This burst of diversification kicked off the Ordovician radiation, the second great wave of increasing marine life on Earth. Around 517 million years ago, the end Permian extinction struck at the same time as major chemical shifts in the oceans and the spread of dark, organic-rich muds. These rocks show that oxygen levels collapsed in mid-depth waters. 2 later crises, the Endrumian and the Furongen events, between about 504 and 489 million years ago, show the same pattern, suggesting that low oxygen zones expanded again and again as the climate shifted and nutrients were recycled through the oceans. Together, these events show that by this time, life and Earth's chemistry were tightly linked. Changes in productivity, oxygen levels, and extinctions were now connected parts of the same system. Recovery after these crises saw the rise of new groups, articulate brachiopods, cephalopods, and the first planktonic graptolites, which would dominate Ordovician ecosystems. The continuity of lineage across such disturbances demonstrates the resilience of the new Cambrian-style food webs.
From Cambrian balance to Ordovician expansion.
As the Cambrian gave way to the Ordovician, the biosphere expanded both in diversity and in depth. Coral-like organisms replaced the extinct archaeocyaths as reef builders. Brachiopods and mollusks spread into cooler, deeper waters. Plankton diversified, transforming ocean chemistry and enhancing carbon sequestration. The slow, steady evolution of these systems turned the episodic Cambrian burst into a sustained geological revolution, the long-term oxygenation and biological modernization of the planet. Cutaway, chemical clues in ancient rocks show that by about 480 million years ago, the deep ocean had gained enough oxygen to stay permanently oxygenated, ending the long history of dark, oxygen-poor deep waters. This change made it possible for more complex food webs to develop in the open ocean and allowed seafloor animals to live at greater depths than ever before. What followed was the Ordovician radiation, a long surge of evolution that lasted until about 445 million years ago and ultimately tripled global biodiversity compared with the Cambrian. As plankton communities became more complex, they grew better at carrying organic material from the surface ocean down into the deep sea. Layers of early Ordovician sediment show repeated episodes when large amounts of this material were buried on the seafloor. Each of these events removed carbon from the atmosphere and helped cool the climate. Together, they show that the changes set in motion during the Cambrian had begun to let life itself influence Earth's temperature, creating a feedback system between biology and climate that still operates today.
The legacy of the Cambrian.
Every subsequent chapter of biological history traces its lineage to the Cambrian. The genetic blueprints written during that explosion have never been erased. All animals, from insects to mammals, bear the imprint of that first experiment in complexity. The trilobite's eye and the vertebrate's skull share the same developmental logic. The coral reef and the Cambrian spongebank operate by identical ecological rules. In the Cambrian, evolution established its permanent vocabulary. Studies of how different animals develop show that the genetic systems used to build limbs in insects, vertebrates, and segmented worms all come from the same ancient ancestor. Evidence from both DNA and fossils places this ancestor right at the beginning of the Cambrian, around 530 million years ago, the time when evolution was at its most inventive. This means the Cambrian explosion wasn't just a burst of new body shapes. It was the moment when the basic genetic toolkit used by all animals today was first locked into place. Geologically, Cambrian sediments comprise some of the planet's richest archives of biogenic carbonate and phosphorite, the enduring record of biology transforming rock. Their isotopic cycles provide the first high-resolution calibration of Earth's biogeochemical rhythms, establishing a synchrony between life, ocean, and atmosphere that continues into the present.
Conclusion: the first age of animals.
The Cambrian explosion ended not with collapse but with integration. In its wake stood a world permanently altered, oxygenated, structured, and inhabited by creatures with eyes to see and skeletons to endure. For the first time, the planet's surface was shaped not only by chemistry and physics but by perception, locomotion, and behavior. The invisible biological foundation built through the Proterozoic had manifested in visible form, and the Earth had entered the age of animals, a world of interaction, predation, and evolutionary conversation that has never ceased. In the next episode, life leaves the shallows. Plants, fungi, and animals begin to colonize the land, and the blue world that birthed them will share its vitality with the continents above.
Episode 9: Life conquers the land, plants, insects, amphibians spread. Part 1: The Ordovician Seas and the Green Revolution. Introduction: The Second Surge.
After the turbulence of the Cambrian, Earth did not fall silent. Around 485 million years ago, the Ordovician period began, bringing the second great expansion of biodiversity. The seas, already busy with trilobites, brachiopods, and mollusks, became richer and more structured. New reefs rose from the seafloor, plankton filled the sunlit waters, and the chemistry of the oceans and atmosphere shifted yet again. Out of this marine abundance came a quieter but world-shaping development, the first true greening of the continents. The Great Ordovician Biodiversification Event, GOBE, between about 470 and 445 million years ago, multiplied known marine genera several-fold. Ordovician rocks record the rise of articulate brachiopods, bryozoans, corals, nautiloid cephalopods, and planktonic graptolites. Geochemical data suggest that global oxygen levels stabilized at roughly half to two-thirds of modern values, enough to sustain large, active animals and intricate ecosystems.
The Ordovician Oceans.
The early Ordovician world was warmer and wetter than before. Gondwana sprawled across the southern hemisphere, while Laurentia and Baltica lay near the equator. High sea levels flooded the edges of continents, creating broad, shallow seas that acted as vast nurseries of life. Sunlight, warmth, and nutrient-rich runoff turned these epicontinental seas into fertile incubators. In these waters, plankton flourished. Radiolarians, siliceous plankton with delicate skeletons, appeared and began to draw silica from seawater. Green algae and early dinoflagellate relatives crowded the upper layers of the ocean, forming the first truly complex planktonic food webs. Below them, oxygenated waters allowed animals to spread into habitats that had been chemically hostile in the Cambrian. Ordovician black shales contain elevated concentrations of redox-sensitive metals such as molybdenum and uranium, while carbon isotope values indicate vigorous burial of organic matter. Together, these signals point to a stable, oxygen-rich, high-productivity oceanic system, the energetic foundation for life's next steps.
The Reef Builders.
Among the most dramatic Ordovician innovations were the first long-lived coral stromatoporoid reefs. Where Cambrian reef builders had flickered and failed, new organisms succeeded. Tabulate and rugose corals, working alongside calcareous sponges and microbial communities, constructed immense reef complexes that stretched for kilometers along tropical shelves. These reefs stabilized sediments, broke waves, and created a labyrinth of caves, ledges, and channels. Reefs transformed marine ecology. They offered shelter from predators, nursery grounds for juvenile animals, and hard surfaces for filter feeders and grazers. Life no longer merely carpeted the seafloor. It now built three-dimensional cities in stone. Within these crowded structures, competition and cooperation drove further diversification. The spread of reef systems intensified the global carbon cycle. Photosynthesis in shallow waters, combined with rapid carbonate deposition, drew atmospheric carbon dioxide down from extreme greenhouse levels. By roughly 460 million years ago, concentrations had fallen enough to begin a long cooling trend that would eventually contribute to Ordovician glaciation.
The green edge, life creeps onto land.
While the seas flourished, subtle changes along the coasts hinted at a new frontier. On damp tidal flats and river margins, thin films of green began to spread. Microscopic algae, already comfortable in intertidal pools, clung to wet rock and mud. In partnership with fungi, they formed sheet-like mats capable of trapping dust and moisture. These were the humble ancestors of all later forests. Spores preserved in mid-Ordovician sandstones showed durable walls and characteristic trilete marks, signatures of embryophytes, true land plants. Chemical biomarkers from the same rocks confirm photosynthetic activity on land, pushing the origin of terrestrial flora tens of millions of years earlier than once thought. Associated fungal remains suggest that lichens and early mycorrhizal partnerships helped these pioneer plants endure desiccation and nutrient-poor substrates.
The atmosphere begins to change.
Though barely visible from afar, these coastal green films began to alter the air itself. Each photosynthetic cell captured carbon dioxide and released oxygen. When storms washed fragments of this biomass into rivers and ultimately the sea, some of that carbon became buried in sediments rather than recycled. Over millions of years, the effect accumulated. Oxygen levels crept upward. Carbon dioxide declined a little more. Enhanced weathering from plant fungus mats also leached phosphorus and other nutrients from bare rock. Washed into the oceans, these nutrients fertilized marine phytoplankton, which in turn drove further organic burial and oxygen release. A feedback loop was underway, land plants and marine microbes working together to reshape the planet's chemistry.
Prelude to a crisis.
The Ordovician prosperity did not last. By 445 million years ago, continued carbon dioxide drawdown and continental configurations favored the growth of ice sheets on Gondwana. Global sea level fell, shallow seas retreated, and reef communities collapsed. Roughly 85% of marine species vanished in the first great mass extinction of the Phanerozoic. Yet the biogeochemical systems forged at this time, the links between land plants, weathering, and climate, survived and would underpin future recoveries.
Part 2. The Silurian Transition, from Water to Land. Introduction. After the ice.
When the Ordovician Ice Age ended around 443 million years ago, glacier melted and seas once again flooded continental margins. Warm, shallow waters welcomed the return of reefs and the diversification of marine life. Trilobites, brachiopods, and corals radiated into newly opened niches, while jawed fishes and giant sea scorpions prowled the water column. Along the shorelines, however, an even more radical transformation was underway. Plants and animals were beginning to build the first true terrestrial ecosystems. The Silurian period, 443 to 419 million years ago, was a time of post-extinction recovery. Coral, stromatoporoid reefs expanded, and vertebrates with jaws and paired fins appeared in abundance. Climate models indicate warm, humid conditions and high but declining carbon dioxide levels, environments well suited to colonization of coastal lowlands by photosynthetic organisms.
The first terrestrial pioneers.
The early Silurian continents were still harsh landscapes, bare rock surfaces, thin protosoils, and pools that dried with the seasons. Yet in sheltered hollows, the first recognizable land plants took hold. These were tiny, leafless stems branching from creeping mats. Each upright shoot ended in a sporangium that released clouds of spores into the air. Filamentous rhizoids anchored the plants to the substrate and helped wick up water. Genera such as Cooksonia and Baragwanathia reveal the anatomical breakthroughs behind this advance. Simple vascular tissues enabled efficient water transport and mechanical support, allowing plants to grow taller than a few millimeters. Even so, early stands rose only centimeters above the ground. The continents were still mostly brown and gray, with scattered patches of green around streams and deltas, but the principle had been established. Photosynthesis could now proceed on land, drawing energy directly from sunlight without the cushion of standing water.
The partnership with fungi and the birth of soils.
The conquest of land depended on alliances as much as anatomy. Beneath the first plants spread networks of fungal hyphae. These mycorrhizal partners penetrated mineral grains, releasing acids that dissolved rock and liberated phosphorus, iron, and trace metals. In exchange, the fungi received sugars from their plant hosts. Together, they formed a single functional unit, capable of exploiting substrates that neither could use alone. As these plant-fungus carpets thickened, they began to create true soils. Rock fragments broke down into clays. Organic matter accumulated as litter and microbial biomass. Pore spaces trapped water and air. Even thin, patchy soils acted as sponges, smoothing out floods and droughts and providing a more stable platform for later vegetation. For the first time, land surfaces hosted self-sustaining ecosystems with internal nutrient cycles.
The invertebrate explorers.
Plants did not remain alone on the Silurian shore. Invertebrates followed, attracted by detritus and algal scum. Millipede-like myriapods, primitive arachnids, and early relatives of springtails crept through the damp litter. Their jointed limbs and wax-coated exoskeletons prevented desiccation and provided support in gravity's full pull. Some grazed on microbial mats, others fed on decaying plant material. Trace fossils, delicate trackways crossing ancient mudflats, show that these animals walked on surfaces exposed to the air rather than submerged in shallow pools. They were the first air-breathing animals, using tracheae to deliver oxygen directly to their tissues. With them, a simple but complete land-based food web emerged: primary producers, decomposers, and small consumers linked by flows of energy and nutrients.
The Silurian air.
With photosynthesis now occurring on land as well as in the sea, the composition of the atmosphere shifted again. Oxygen levels rose to perhaps a fifth of modern concentrations. Carbon dioxide continued its slow slide. Lightning, storms, and even the first wildfires responded to this changing mixture of gases. By the end of the Silurian, Earth's atmosphere had acquired a modern-like oxidative potential. The continents were still only lightly greened, but the stage was set for much larger organisms to thrive in open air.
Part 3: The Devonian World, Forests, Fishes, and Amphibians. Introduction: The Age of Green and Bone.
By the dawn of the Devonian, around 419 Ma, the balance between land and sea had changed once more. The continents, especially in equatorial regions, now carried extensive belts of vegetation. River valleys supported dense mats of plants, early insects scuttled among stems, and freshwater fishes filled the streams. The Devonian is often called the Age of Fishes, but it was just as much the age of the first forests and the first-limbed vertebrates.
The rise of forests.
Earlier plants had hugged the ground, limited by weak tissues and simple plumbing. During the Devonian, they evolved lignin-reinforced cell walls, complex vascular systems, and branching trunks capable of supporting large crowns of foliage. The earliest trees, such as Archaeopteris, reached heights of 10 meters or more. Their woody stems, deep roots, and shedding fronds created the world's first true forests, shaded, humid environments radically different from the open Silurian lowlands. Forest growth transformed Earth's surface. Roots stabilized riverbanks and encouraged rivers to meander rather than braid. Thick leaf litter accumulated on the ground, feeding fungi and invertebrates and building deep, organic-rich soils. Lignin-bearing wood decayed slowly, and much of it ended up buried in sediments, locking carbon away for geological timescales. As a result, atmospheric carbon dioxide fell and the climate cooled.
The Age of Fishes.
While forests conquered the land, the oceans and rivers underwent their own revolution. Fishes diversified into a spectacular array of forms. Heavily armored placoderms dominated many marine ecosystems, including giants such as Dunkleosteus with powerful biting plates. Cartilaginous fishes explored the open seas, while bony ray-finned and lobe-finned fishes experimented with new body plans and modes of life. Among the lobe-fins, evolution was quietly preparing for a move onto land. Their paired fins contained sturdy internal bones, precursors of humerus, radius, and ulna. Some developed lungs and nostrils that allowed them to breathe air in oxygen-poor waters. Fossils such as Eusthenopteran and Tiktaalik capture intermediate stages in this transition. Fish with limb-like fins, mobile necks, and flattened skulls suitable for life in shallow streams and swamps.
The insects take wing.
On land, arthropods diversified alongside the plants. By the mid-Devonian, the first true insects appeared, some already possessing wings. These early flyers exploited the still air above forests, becoming the first animals to claim the aerial realm. Their appearance opened an entirely new dimension of ecological interaction, from spore and pollen transport to aerial predation, and further tied the fate of land plants to that of animals.
The amphibian frontier.
Late in the Devonian, some lobe-finned fishes took the final steps onto land. Seasonal droughts, crowded swamps, and unstable shorelines favored animals that could haul themselves from pool to pool or lie in ambush at the water's edge. From these pressures emerged the first tetrapods, vertebrates with digits instead of fin rays. Acanthostega and Ichthyostega, known from Greenland, carried limbs with multiple digits, robust vertebral columns, and lungs supported by rib cages. They still retained internal gills and fish-like tails, and probably spent much of their time in shallow water, but they could walk or crawl on solid ground. Trackways from Poland suggest that tetrapod-like animals were experimenting with terrestrial locomotion even earlier. For the first time, vertebrates joined plants and invertebrates in the continental realm.
The new web of life.
By the end of the Devonian, terrestrial ecosystems had become richly interconnected. Forest canopies intercepted rainfall and fed complex river systems. Soils regulated water flow and recycled nutrients. Insects and other arthropods fed on plants and each other. Amphibians hunted along swamp margins and forest floors. Fires periodically swept through dry seasons, clearing vegetation and opening space for regrowth. The continents themselves now pulsed with the full rhythm of life: photosynthesis, respiration, decay, and predation. To an observer watching this transformation in fast forward, the Ordovician to Devonian would look like a slow but relentless tide of life moving out of the ocean. First a faint green stain along shorelines, then scattered tufts on riverbanks, then low carpets creeping inland, and finally dark belts of forest following the paths of water across the continents. At every step, animals followed. Tiny arthropods first, then insects and amphibians, each wave of colonists testing the limits of air, gravity, and dryness. Seen from space, the changes would be just as dramatic. The blue-white planet of the early Paleozoic acquired swirls of brown and green along its coasts, then deep interior smudges where forests took root on upland plateaus. Seasonal cycles intensified as vegetation altered how continents absorbed and released heat. Clouds gathered differently over wooded regions, and rainfall patterns shifted in response. In this sense, the Devonian did more than add life to land. It turned the continents into dynamic parts of the climate system rather than passive slabs of rock. For evolution, the opening of the terrestrial realm was like adding extra dimensions to a game board. Animals and plants that had once interacted only in two-dimensional seafloor communities suddenly had access to height, tree trunks, canopies, burrows, and riverbanks. New ecological roles appeared: canopy browsers, forest floor scavengers, ambush predators waiting at the water's edge, aerial hunters pursuing winged prey. The diversification of form and behavior that followed would, over the next 100 million years, give rise to ecosystems recognizably similar in structure to those of today, even if their cast of species was entirely different.
Part 4: Legacy and Climate Crisis. The Late Devonian Extinction. Introduction: The greenhouse unravels.
Yet the Devonian triumph contained the seeds of crisis. As forests spread and soils deepened, weathering rates soared. Rivers carried unprecedented loads of nutrients into coastal seas. There, plankton blooms flourished and then died, their decomposition stripping oxygen from bottom waters. At the same time, the drawdown of atmospheric carbon dioxide cooled the climate, encouraging glaciation on Gondwana. The ocean, once the secure cradle of complex life, began to falter.
The geochemical engine of collapse.
The late Devonian extinction, unfolding between about 375 and 359 million years ago, was not a single blow but a drawn-out sequence of crises. Widespread deposition of organic-rich black shales records repeated intervals when large regions of the ocean became anoxic or even euxinic, devoid of oxygen and loaded with hydrogen sulfide. Reef systems collapsed, many placoderm fishes disappeared, and long-stable trilobite families vanished. Two pulses stand out. The Kellwasser event, around 372 million years ago, devastated reef communities. The later Hangenberg event, near the Devonian Carboniferous boundary, eliminated many remaining fish lineages and reshaped vertebrate evolution. Behind both lay the same engine: nutrient-choked, oxygen-starved seas linked to the success of land plants and the cooling of the climate.
Oxygen, fire, and survival.
Paradoxically, while the ocean suffocated, the air above them may have become highly oxygenated. Extensive forests and the burial of their remains drove atmospheric oxygen to levels comparable to or perhaps exceeding modern values. Charcoal fragments in late Devonian rocks show that wildfires became frequent features of the landscape. These fires reduced forest cover, accelerated soil erosion, and supplied yet more nutrients to the oceans, tightening the feedback loop that underpinned the crisis. Life, however, endured. On land, plant, and insect diversity rebounded within a few million years. In the seas, new reef builders and fish groups emerged. The surviving lobe-finned vertebrates would give rise to amphibians, reptiles, and ultimately mammals. The catastrophe acted as an evolutionary filter, pruning some branches while allowing others to flourish.
Conclusion: the living continents.
By the close of the Devonian, Earth's continents had been permanently transformed. What began in the Ordovician as microscopic green films on damp rock had become forests that reshaped rivers, soils that altered climate, and animal communities that filled land, water, and air. Plants, insects, fishes, and early amphibians together created a fully integrated biosphere in which atmosphere, hydrosphere, and lithosphere were tightly bound by life. The price of this progress was instability. The same feedbacks that cooled the planet and enriched the air could, when pushed too far, suffocate the seas and trigger extinction. Yet through these crises, the planet learned to regulate itself more effectively. Weathering, climate feedbacks, and biologically driven carbon cycles, tested in the Devonian, would keep Earth broadly habitable for the rest of its history. Above all, these Paleozoic innovations established principles that still govern the modern world. Plants linked rock to air through roots and leaves. Fungi and microbes closed the loop by returning nutrients from dead tissue to the soil. Animals stitched habitats together by moving energy and matter across boundaries. Once these feedbacks were in place, later radiations of reptiles, dinosaurs, birds, and mammals could unfold on a planet whose basic operating rules had already been set by mosses, club mosses, and the first forests of the Devonian. In the next episode, the story will continue into the Carboniferous and Permian, ages of coal forests, giant insects, and the first true reptiles, where the greening of the continents reaches its zenith before another reckoning reshapes the world again.
Episode 10: The Great Dying, Permian extinction wipes out 90% of species. Part 1: The Carboniferous World, Forests, Fire, and Oxygen. Introduction: A planet of forests.
By 359 million years ago, the Carboniferous period opened on a planet draped in green. From Devonian survivors arose vast lowland forests across the equatorial supercontinent Euramerica. Tree ferns, lycopsids, horsetails, and seed ferns rooted in deep, waterlogged soils, their crowns knitted into canopies stretching for hundreds of kilometers. The air grew dense with moisture and, increasingly, with oxygen. For the first time, the continents functioned as a planetary lung. Forests drew down carbon dioxide, buried organic matter, and exhaled oxygen into the sky. During the Carboniferous period, about 359 to 299 million years ago, Earth went through two main chapters called the Mississippian and Pennsylvanian. At this time, carbon dioxide in the air dropped to very low levels, while oxygen rose to around 1/3 of the atmosphere, the highest it has ever been. Huge amounts of plants were buried in swampy wetlands, locking away carbon and causing this change. As a result, the air became thicker, which slightly affected how animals breathed, how fires burned, and even how the climate worked.
The coal forests.
Equatorial regions were dominated by swamp forests, labyrinths of standing water, floating peat, and towering lycopsid trees such as lepidodendron and sigillaria, some over 40 meters tall. Their trunks bore diamond-patterned scars. Their roots formed dense underground networks in waterlogged soils. Beneath the canopy, shade-tolerant plants thrived amid pools, fallen trunks, and decaying litter. Death in these wetlands did not mean rapid decay. Fallen trees and leaves sank into anoxic bogs where bacteria worked slowly. As sediments accumulated, thick peat layers formed. Over millions of years, burial, heat, and pressure compressed these peats into coal, vast, black archives of a world ruled by plants. In the Pennsylvanian period, some coal layers grew to be as much as 30 meters thick, far larger than anything we see forming today. These layers often appear in repeating patterns with sandstone, showing that sea levels were rising and falling as the planet shifted between ice ages and warmer times, guided by changes in Earth's orbit. The chemistry of nearby limestone rocks reveals that huge amounts of carbon were locked away underground, which helped oxygen in the atmosphere steadily climb higher.
Giants of air and swamp.
Oxygen-rich air transformed animal possibilities. Arthropods, whose respiration depends on diffusion through tracheae, achieved sizes impossible today. The skies held dragonflies like Meganeura with wingspans approaching 70 centimeters. The forest floor hosted Arthropleura, millipede-like giants over 2 meters long. Amphibians, liberated by abundant wetlands, diversified into a wide range of body plans, from slender insect eaters to bulky semi-aquatic predators. Among them appeared a new kind of vertebrate, small, lizard-like reptiles that laid amniotic eggs protected from desiccation. These early amniotes could reproduce away from open water, creeping into drier microhabitats at forest edges and uplands. When oxygen made up about 35% of the air, insects could grow up to 10 times larger than the biggest ones alive today, which matches the size of the largest insect fossils we found. Around the same time, early reptiles like Hylonomus, about 312 million years ago, had strong legs for walking on land and eggs with tough, leathery shells. These features marked a key step in evolution that eventually gave rise to reptiles, birds, and mammals.
The Fire Age.
High oxygen had a cost. Vegetation ignited easily. Lightning strikes over humid forests sparked frequent wildfires. Flames raced through canopy and undergrowth, leaving behind charcoal-rich ash layers. Fire became a central ecological process, clearing old growth, recycling nutrients, and shaping plant evolution. Species with thick bark, rapid regrowth, or spore releases timed to follow fires prospered. In the Carboniferous period, many coal deposits contain 5 to 15% fossil charcoal, evidence of ancient wildfires. Experiments show that when oxygen in the air rises above about 25%, fires can keep burning on their own. The large amount of charcoal in these rocks tells us that oxygen was very high back then, and that frequent fires help stop oxygen from climbing even higher by burning plant material and sending carbon dioxide back into the atmosphere.
Climate cycles and ice.
While the tropics teemed with life, the southern supercontinent Gondwana lay under extensive ice sheets. The Carboniferous climate oscillated between glacial and interglacial states, driven by orbital cycles and continental geometry. During glacial maxima, sea levels fell and coastal swamps shrank. During interglacials, meltwater flooded lowlands and peatlands expanded. The rock record preserves these pulses as alternating marine and terrestrial layers, the heartbeat of a forested, glaciated world. Glacial deposits from South Africa, South America, and Australia date between 335 and 295 Ma, confirming a prolonged Gondwanan ice age. Oxygen isotope shifts of up to 2 per thousand correspond to global temperature swings of several degrees, in tune with 100,000-year eccentricity cycles. Coal-forming wetlands waxed and waned with these sea level changes, coupling ice volume to oxygen and carbon cycles.
The end of the coal forests.
In the late Carboniferous, tectonic drift drew continents together. As Pangaea assembled, mountain building reshaped atmospheric circulation. Interior basins dried, monsoons weakened, the equatorial wet belt narrowed and fragmented. Coal forests that had once formed continuous belts across Euramerica and beyond retreated into refuges along coasts and river plains. Dryer-tolerant seed ferns and primitive conifers replaced swamp flora on uplands. Around 305 million years ago, Earth's rainforests suddenly broke down. We can see this because coal, which forms from dense plant life, stopped building up, and the variety of plant spores dropped sharply. At the same time, chemical signals in the oceans show that less plant material was being buried, which marked a turning point in the amount of oxygen in the air. Rising mountain ranges like the Appalachians and Veriskans changed wind and rainfall patterns, slowly breaking apart the wet, lush climate system that had kept oxygen levels so high during the Carboniferous period.
Conclusion: the peak of breath.
The Carboniferous was Earth's great experiment in oxygen. Its forests buried staggering amounts of carbon and pushed oxygen to levels never again reached, allowing giant arthropods, extensive fires, and dense swamp ecosystems. Yet this abundance depended on a delicate interplay of climate, sea level, and tectonics. As continents fused and climates dried, the wetland engine faltered. The age of forests drew down its own curtain, and a new, harsher world emerged, the stage on which the Permian drama would unfold.
Part 2: The Permian Transformation. Continents unite, climates dry. Introduction: A changing planet.
As the Carboniferous ended, Earth's geography and climate reorganized. Over tens of millions of years, collisions between continents created the supercontinent Pangaea, a single landmass stretching from pole to pole and spanning much of the globe. Its vast interior lay far from the moderating influence of oceans, fostering seasonal extremes and immense deserts. The humid, coal-forming forests shrank to coastal fringes and highlands. Seed plants with drought-resistant traits, conifers, cycads, ginkgos, and glossopterids, spread over upland plains. Meanwhile, atmospheric oxygen declined and carbon dioxide rose. The Permian world was biologically sophisticated but environmentally unstable. The Permian, 299 to 252 million years, saw carbon dioxide rebound from under 1,000 ppm to around 2,000 ppm, while oxygen fell from roughly 30% to near 15%. Charcoal content in coals plummets during this interval, indicating reduced fire frequency and lower oxygen. Global temperatures climbed to averages near 28 degrees Celsius, with polar regions largely ice-free. The consolidation of continents also altered ocean circulation, predisposing basins to stagnation and anoxia.
Pangaea, the supercontinent.
Pangaea's assembly was one of the great tectonic revolutions. The closure of multiple oceans raised a continuous chain of mountains from North America through Europe into Asia. These ranges diverted wind belts and monsoons, generating hyperarid interiors. Seasonal temperature swings of 40 degrees Celsius or more likely characterized inland plateaus, while coastal margins experienced intense, sometimes erratic rainfall. Orogenic belts such as the Appalachians, Variscans, and Urals record collision between continental blocks between 320 and 270 million years ago. Climate simulations show that such a configuration produces strong monsoonal cells around Pangaea's margins and vast subtropical deserts in its center. Dust from these drylands, preserved as red beds and loess, testifies to frequent storms and long-distance sediment transport. The closure of seaways also restricted deep water formation. Without vigorous polar sinking, the oceans circulated more slowly. Deep basins became prone to oxygen depletion, and black shales with pyrite and organic matter accumulated even before the final crisis. The marine world was becoming stratified, thin oxygenated surface layers floating above expanding oxygen-poor depths.
The new flora and fauna.
The shift to a drier climate drove a turnover in life. The old coal forest flora of lycopsids and tree ferns declined. In their place, seed plants flourished. In the south, glossopterid forests spread across Gondwana. In the north, conifers and ginkgos dominated uplands. Their seeds and tough leaves allowed survival through seasonal drought and cold. On land, reptiles seized the initiative. Amphibians, tied to water for reproduction and vulnerable to desiccation, retreated to moist refuges. Synapsids, the mammal-like reptiles, diversified into herbivores with deep bodies and broad teeth, and predators with large skulls and sailbacks for thermoregulation. Later synapsids, the therapsids, evolved differentiated teeth, more upright limbs, and higher metabolic rates, foreshadowing the mammalian condition. Glossopterid fossils, with distinctive tongue-shaped leaves, occur across India, Australia, South Africa, and Antarctica, providing key evidence for Gondwana's former unity. Synapsids such as Dimetrodon and Edaphosaurus in the early Permian, and therapsids like Mosechops and Gorgonops later, show a trend toward more active, endotherm-like physiology, an adaptation to the wide ranges and harsh climates of Pangaea.
The desert kingdoms.
By mid-Permian time, Pangaea's interior supported vast dune fields, salt flats, and ephemeral rivers. Wind-blown sands piled into dunes hundreds of meters high. Seasonal floods carved temporary channels and left behind mud-cracked flats. Insects evolved tougher exoskeletons. Beetles and cockroach-like forms were well-suited to aridity. Reptiles with scaly skin and efficient kidneys thrived where amphibians could not. The Coconino sandstone in North America and equivalent redbed formations in Europe preserve large cross-bedded dunes and trackways of reptiles moving confidently across dry sand. Interbedded evaporites, halite, gypsum, and hydrite indicate cycles of lake filling and complete desiccation. These deposits document a supercontinent whose heartland was as dry as any desert in later Earth history.
The oceans of the Permian.
Around Pangaea spread the enormous Panthalassic Ocean and subsidiary basins like the Paleotissese. With little polar ice and restricted circulation, these waters warmed and stratified. Life flourished in some shallow shelves, where reefs of sponges, algae, and early corals developed, but deeper zones increasingly slipped into anoxia. Permian marine sections in China, Russia, and North America contain black shales with fine pyrite and enriched trace metals, diagnostic of low oxygen conditions. The turnover from brachiopod-dominated faunas to more resilient bivalves and ammonoids reflects adaptation to these stressed environments. Carbon isotope swings of several per mil suggest a carbon cycle already prone to disruption before the final catastrophe.
Decline of the oxygen world.
As coal swamps vanished and previously buried organic matter was uplifted and oxidized, the oxygen surplus bled away. Fire frequency dropped, charcoal became rare, giant insects disappeared. Animals adapted to thinner, less oxygen-rich air, respiratory surfaces expanded, and thoracic cavities enlarged. Inertinite content in late Permian coals falls below 3%, consistent with oxygen near or below 15%. Nitrogen isotope data indicate reduced oxidative processes in soils and waters. Insects shrink in maximum size, and vertebrate fossils show trends toward more efficient breathing structures, a physiological response to an atmosphere that no longer offered superabundant oxygen.
Conclusion: the fragile equilibrium.
By the late Permian, Earth seemed to have reached a new equilibrium, a unified continent, sophisticated plant and animal communities, and a climate generally warm but capable of regional extremes. Yet under this apparent stability lay vulnerabilities: stagnant oceans, declining oxygen, and a carbon cycle easily perturbed. The biosphere that had once cooled the planet and filled it with breath was losing its tight control over chemistry. It would take only one additional shock to tip this system into chaos.
Part 3: The catastrophe, the end Permian mass extinction. Introduction: the breath of the Earth fails.
Around 252 million years ago, the long balance of the Paleozoic biosphere broke. For over 100 million years, forests, reefs, and deserts had exchanged gases with the sky in a relatively stable rhythm. Then, in a geologic instant, that rhythm collapsed. A flood of volcanic fire in Siberia injected unprecedented amounts of carbon and toxins into the atmosphere. Oceans warmed, stagnated, and turned poisonous. Land surfaces were scoured by acid rain and heat. In this brief interval, about 90% of marine species and 70% of terrestrial vertebrate genera vanished. The Paleozoic ended in a single, devastating Great Dying. High-precision UPB dating of boundary layers, especially at Meishan in China, shows that the main extinction pulse lasted less than 60,000 years. Fossil counts indicate an order-of-magnitude drop in marine diversity. This was not a slow decline but a sharp cliff in the history of life.
The Siberian Traps.
The immediate trigger was the eruption of the Siberian Traps, a large igneous province covering several million square kilometers. Over roughly a million years, great fissure eruptions poured lava across northern Pangaea. Each pulse released carbon dioxide, sulfur gases, and halogens. Lava intruding into coal and carbonate beds baked additional greenhouse gases out of the crust. Estimates of total lava volume exceed 3 to 5 million cubic kilometers. If each erupted batch released carbon dioxide at rates comparable to modern flood basalts, cumulative emissions easily surpassed 10,000 gigatons of carbon. Contact metamorphism of underlying coals and evaporites may have added thousands of gigatons more, including methane, a far more potent greenhouse gas. Mercury spikes and sulfur isotope anomalies in boundary sediments record repeated episodes of intense degassing, acid rain, and stratospheric damage.
Runaway greenhouse.
The climatic response was swift. Atmospheric carbon dioxide soared to many times pre-eruption levels. Global temperatures climbed by 8 to 10 degrees Celsius. Tropical seas warmed to near lethal temperatures. Land surfaces in equatorial Pangaea approached 35 to 40 degrees Celsius, stressing plant and animal physiology. Rain became acidic, stripping soils and releasing metals and nutrients into rivers and coastal waters. Ocean acidification dissolved shells and undermined carbonate-producing organisms. At this boundary in Earth's history, evidence from tiny fossil teeth shows a sudden jump in temperature. At the same time, chemical clues from ancient rocks reveal that the oceans became more acidic, with pH levels dropping close to 7. This made the water nearly unable to hold the minerals needed for shells. For many creatures that built shells, the environment crossed a critical line. They could no longer grow or survive.
The poison seas.
As the climate warmed and more nutrients flowed into the oceans, the water became layered. The surface stayed warm and held oxygen for a while, but the deeper layers stopped mixing and lost oxygen. Dead plants and animals sinking down made this worse, and bacteria that produced toxic gases spread. Eventually, waters filled with hydrogen sulfide, poisonous to most life, rose up into shallow seas and even into the sunlit surface zone. Black shales at the Permian-Triassic boundary show iron speciation signatures and tiny pyrite framboids typical of Euxinia. Biomarkers such as isorenirotane, produced by green sulfur bacteria, demonstrate that sulfide-rich waters extended into the photic zone. Models suggest that hydrogen sulfide outgassing could have damaged the ozone layer, letting UV radiation further stress life on land and in shallow seas. Reefs collapsed first, followed by brachiopods, corals, crinoids, trilobites, and many plankton. Food webs unraveled from top and bottom simultaneously. Predators lost prey. Primary producers lost habitable niches. In some regions, only a few opportunistic species clung on in the aftermath.
Collapse on land.
On land, the same forces wreak havoc. Acid rain and heat killed forests, leaving bare soils vulnerable to erosion. Dust storms scoured plains. Rivers carried silt, metals, and nutrients into already stressed coasts. Glossopterid and conifer forests disappeared. Fern-dominated floras briefly expanded into the gaps, a disaster flora signal seen worldwide. Herbivorous synapsids lost their plant food. Carnivores starved as prey dwindled. Only small, generalist, often burrowing animals had the resilience to survive. In South Africa's Karoo Basin, vertebrate assemblages shift from diverse communities of large Decinodonts and Gorgonoptions to impoverished faunas dominated by the small burrowing Lystrosaurus. Bone histology shows growth interruptions and signs of stress. Pollen and spore records reveal sharp turnovers, including global fern spikes indicative of rapid colonization of devastated landscapes by hardy plants.
The final pulse.
In the global rock record, the peak of the catastrophe corresponds to a thin, dark layer rich in soot, ash, and chemical anomalies. Carbon isotope values plunge by 6 to 8 per thousand, reflecting the oxidation of vast stores of organic and methane carbon. Extinction peaks coincide with maximum warming, most severe anoxia, and strongest geochemical shifts. At Meishan in China, scientists study a thin rock layer less than a meter thick that captures signs of a global disaster. It shows unusually high mercury from massive volcanic eruptions, chemical shifts that reveal a collapse in plant and animal life, and strange patterns in elements like selenium and sulfur that suggest Earth's normal cycles broke down almost completely. After this point in time, many types of ancient life from the Paleozoic era disappeared forever.
Conclusion: the long night.
The end Permian extinction was more than a spike in volcanic activity. It marked a moment when feedbacks that had kept climate and chemistry within habitable bounds were overwhelmed. Carbon, once cycled by life in stabilizing loops, surged uncontrollably through atmosphere and ocean. Heat, acidification, and anoxia pushed Earth close to biological reset. Yet even here, life did not vanish completely. A few lineages endured through the worst conditions the planet has seen since complex life evolved, setting the stage for a slow and radical recovery.
Part 4: Aftermath and Recovery, the Triassic Dawn. Introduction: a world of ash and silence.
When volcanic fury waned and the climate began to stabilize, the world that remained was unrecognizable. Many landscapes were stripped to bare rock and thin soils. Rivers ran with chemically loaded waters. Seas were murky, acidic, and still oxygen poor. For hundreds of thousands of years, ecosystems were dominated by disaster taxa, hardy, opportunistic organisms that could tolerate extreme conditions. Early Triassic sediments frequently show disaster facies, monotonous sandstones and shales with low diversity, often dominated by a single bivalve or brachiopod genus. Carbon isotope values remain depressed for several 100,000 years, indicating a carbon cycle still far from equilibrium. Trace metal proxies show that deep ocean anoxia persisted for at least 2 million years after the main extinction pulse.
The survival biota.
In the oceans, thin-shelled bivalves, simple ammonoids, and microbial mats colonized seafloors largely devoid of complex communities. On land, a few synapsids, notably Lystrosaurus, and tough ferns and seed ferns spread across devastated terrain. These survivors, not particularly impressive in size or specialization, had the crucial advantage of resilience, fast reproduction, generalist diets, and tolerance of heat and hypoxia. In some earliest Triassic formations, Lystrosaurus fossils make up more than 80 to 90% of vertebrate remains. In marine strata, Chlorea bivalves occur in dense, nearly monospecific beds. Such dominance patterns reveal ecosystems simplified to a few robust forms, with most ecological roles temporarily vacant.
The long recovery.
Recovery from the Great Dying was slow and uneven. For millions of years, climatic and chemical instability persisted, with secondary carbon pulses and smaller extinction events interrupting rebounds. Yet gradually, diversity returned. Marine ecosystems saw the rise of new groups: modern-style corals, scleractinians, neogastropods, and diversified ammonoids. On land, synapsid survivors gave rise to cynodonts and early mammal relatives, while reptiles radiated into new forms. Recovery from the Great Dying was slow and uneven. For millions of years, climatic and chemical instability persisted, with secondary carbon pulses and smaller extinction events interrupting rebounds. Yet gradually, diversity returned. Marine ecosystems saw the rise of new groups: modern-style corals, scleractinians, neogastropods, and diversified ammonoids. On land, synapsid survivors gave rise to cynodonts and early mammal relatives, while reptiles radiated into new forms. By about 247 to 240 million years, carbon isotope curves trend positive and stabilize, indicating renewed organic burial and a rebalanced carbon cycle. New coral reefs appear, and trace metals suggest improved ocean ventilation. Biodiversity studies show that it took roughly 5 to 10 million years for ecosystems to regain moderate complexity, and up to 30 million years to reach pre-extinction global diversity levels.
The new atmosphere and climate.
As vegetation re-established itself, the terrestrial carbon and oxygen cycles rebooted. Weathering of volcanic rocks and uplifted sediments drew down carbon dioxide. Soils thickened. Charcoal returns to the rock record, signaling a rebound in oxygen and the comeback of fire. Climate remained warm, especially at low latitudes, but extremes softened compared with the crisis peak. Monsoonal patterns over Pangaea generated belts of forest, savanna, and desert. Rocks from the Middle Triassic show signs that Earth's air had plenty of oxygen, even more than today. Charcoal remains suggest frequent fires, which need high oxygen to burn. Coral reefs from this time record tropical seas about 30 degrees Celsius and normal ocean chemistry, clear evidence that the oceans had bounced back from earlier acidification and oxygen loss.
The rise of the archosaurs.
On this reconstructed stage, new rulers emerged. Among reptiles, the archosaurs, with upright limbs, efficient lungs, and high aerobic capacity, radiated into predators and herbivores across Pangaea. From them would later evolve crocodiles, pterosaurs, and dinosaurs. Synapsid survivors diversified too, producing cynodonts with more mammal-like jaws, teeth, and perhaps fur. The Triassic became a laboratory for modern ecosystems. Fossil assemblages from Triassic basins in South America, Europe, and China show the rapid spread of archosaurs within 10 to 20 million years of the extinction. Early dinosaurs appear by the late Triassic, while mammaliform synapsids arise from cynodont ancestors. The Great Dying thus cleared ecological space that these new clades would fill, reshaping the trajectory of vertebrate evolution.
A planet reforged.
By the end of the Triassic, the scars of the Great Dying were obscured by new growth. Coral reefs once again ringed tropical coasts, conifer forests cloak highlands, river valleys supported diverse faunas of reptiles and early mammals. The biosphere was not a restored Paleozoic world but a fundamentally new one, built on different players and relationships. The Mesozoic Earth, the world of dinosaurs, early mammals, and modern-style oceans, arose from the ashes of the Permian. Geochemical models suggest that total organic carbon burial during the Triassic exceeded the losses from the extinction, effectively resetting the long-term carbon and oxygen cycles. The supercontinent Pangaea remained in place, but under a climate and biosphere that had learned to operate under different rules.
Conclusion: Life's second beginning.
The Great Dying stands as the most severe test life has faced since it began. It shows that the biosphere and planet are coupled in a feedback system that can, under extreme forcing, spin into catastrophe. Yet it also shows that life is tenacious. From a near-sterile world, evolution rebuilt complexity, inventing new forms and ecologies. Warm-bloodedness, more efficient lungs and circulation, and new reproductive strategies all flourished in the post-crisis world. The Permian extinction thus marks both an end and a beginning, the final chapter of the Paleozoic and the prologue to the age of dinosaurs and mammals. Earth had passed through fire, poison, and near suffocation, and emerged into a new era, carrying forward the lessons of its greatest dying.